So, here's something worth thinking about as you settle in tonight. Right now, at this very moment, there is a security operation running inside your body that makes anything humans have ever built look like a child's toy. Millions of cells are on patrol. They're circulating through your blood, drifting through your tissues, stationed at every border where the outside world meets your insides, your skin, your lungs, the lining of your gut, the membranes of your nose and throat. And every single one of them is doing the same thing over and over, without rest, without pause, without you ever noticing. They're checking. They encounter a molecule and they ask a question. Is this one of ours? Or isn't it? Most of the time the answer is simple. Ours, ours, ours. Move along. And you never feel a thing. You don't notice the bacteria that landed on your lip when you touched your face an hour ago. You don't notice the fungal spore you inhaled walking past a garden this afternoon. You don't notice the tiny fragment of viral protein that brushed against the lining of your throat on the subway. Your security system handled all of it. Quietly, efficiently, without sending a memo up to your conscious brain. You just went on with your day, completely unaware that your body had been fighting and winning dozens of tiny skirmishes since you woke up this morning. And here's what makes the story of vaccines so fascinating and so much stranger than most people realize. A vaccine doesn't fight disease for you. That's the part people sometimes get a little fuzzy on, and it's worth getting very clear about right at the start. A vaccine doesn't kill a pathogen. It doesn't neutralize a toxin. It doesn't attack anything at all. What it does, and this is so much more elegant than any drug or antibiotic or chemical weapon we've ever invented, is teach. It teaches your body's own defense system to recognize a threat that it hasn't met yet. Think of it this way. Imagine you're the head of security at a building, and someone hands you a photograph. Here's a person who might try to break in next week. Study the face. Memorize it. You don't have to do anything right now, but when that face shows up at the door, the recognition should be instant, and the response should be overwhelming. That's the core idea behind every vaccine ever made, from the very first crude attempts thousands of years ago to the molecular engineering marvels of the 21st century. Show the immune system a photograph of a threat. Let it study the image. Let it memorize the face. And then wait. But here's where the questions start to pile up, and they're big questions, the kind that scientists spent centuries trying to answer, and in some ways are still answering today. How does your body learn to recognize something it has never encountered before? What's actually happening inside your cells when a vaccine goes to work? How did humans figure out, long before they understood any of the biology, that you could train the body to defend itself against diseases that would otherwise kill 1 in 3? And why, after more than 200 years of vaccine science, do some vaccines protect you for a lifetime, while others fade in a matter of months? In 1803, to get a vaccine across the Atlantic Ocean, you needed a ship full of orphan children, infecting them one by one, arm to arm, to keep the virus alive during the voyage. In 2020, to get a vaccine to the other side of the world, someone sent an email. How did we get from there to here? And what does the answer tell us about the most sophisticated defense system that evolution has ever produced? The one that's working inside you right now, as you lie here, as you breathe, as you begin to let the day go. That's what we're going to spend tonight exploring. And there's no rush at all. Let's start where the story really starts, which is with the security system itself. Because before you can understand how a vaccine teaches your body, you need to understand what your body already knows. Let's begin with something that might surprise you. Your body was born knowing how to fight. Not everything, not with precision, not with the kind of targeted, devastating accuracy that comes later, but from the moment you took your first breath, possibly even before, you had defenses in place. Ancient defenses. Defenses that evolved over hundreds of millions of years, long before anything resembling a human being walked the earth. Scientists call this your innate immune system. And the word innate is important. It means inborn, built in, present from the start, no training required. Think of it as the first ring of security around your building. These are the guards who don't need photographs. They don't need to know who the intruder is specifically. They just need to know that something doesn't belong. And they're remarkably good at that general kind of recognition, because the things that don't belong tend to have certain telltale features, certain markers that your cells have learned to spot over the course of deep evolutionary time. Take bacteria, for example. Bacteria are very different from your own cells. Most bacteria have a rigid outer wall made of a substance called peptidoglycan, and your cells don't have anything like it. It's foreign, it's distinctive. It's like walking into a building wearing a uniform that nobody inside would ever wear. Your innate immune system has receptor molecules, proteins on the surface of certain cells that are specifically shaped to recognize components like peptidoglycan. When these receptors grab onto that bacterial wall material, it's an instant alarm. Something is here that shouldn't be. These receptors have a wonderful name. They're called pattern recognition receptors, and the most famous family of them are the toll-like receptors, which were discovered in the 1990s and earned Jules Hoffman and Bruce Beutler a Nobel Prize in 2011. The name toll-like comes from a gene first found in fruit flies, because sometimes the most fundamental discoveries about human immunity come from studying insects, which, it turns out, share some of the same ancient defense mechanisms we do. That tells you something about how old these systems are. When fruit flies and humans share the same basic security hardware, you're looking at something that was already working hundreds of millions of years ago, before the evolutionary paths that led to insects and mammals diverged. So, your innate immune system recognizes general patterns. Bacterial wall components, double-stranded RNA, which is a hallmark of many viruses, because your own cells normally use single-stranded RNA. Flagellin, the protein that makes up the whip-like tails bacteria use to swim around. These are called pathogen-associated molecular patterns, and they're essentially the uniforms of the microbial world. Your body doesn't need to know which specific bacterium has shown up. It just needs to know that a bacterium has shown up, and then it responds. The response is fast and brutal, and honestly, kind of impressive. Within minutes of detecting an intruder, your innate system can deploy several different types of cells to deal with the problem. There are neutrophils, which are the most abundant white blood cells in your body, and they are essentially professional killers. They arrive at the site of infection quickly, and their main talent is engulfing bacteria and destroying them. They swallow the pathogen whole, trap it inside a compartment called a phagosome, and then flood that compartment with toxic chemicals, reactive oxygen species, enzymes that break down proteins, antimicrobial peptides. The bacterium is digested,
dissolved, gone. Then there are macrophages, which are larger, longer-lived cells that do something similar, but with an added twist that will become very important later in our story. Macrophages don't just eat pathogens. After they've digested an intruder, they take fragments of it, little pieces of the pathogen's proteins, and display those fragments on their own surface. They hold them up, almost like showing a badge to other cells in the area. Look at this. Look what I found inside. This is what's in here. And that display, that act of holding up a fragment of the enemy for others to see, is one of the most important things that happens in your entire immune response. We'll come back to it. Remember it.
Your innate system also has chemical weapons. Proteins called interferons, which infected cells produce and release into their surroundings as a warning signal. The name says it all. Interferons interfere with viral replication. When a cell detects that it's been infected by a virus, it can release interferons that tell neighboring cells to ramp up their antiviral defenses, essentially putting the whole neighborhood on alert. And there's something else your cells can do when they realize they've been compromised. Something that sounds almost noble in a grim sort of way. They can kill themselves. Deliberately. In a controlled, orderly fashion. This process is called apoptosis, and it's nothing like the chaotic rupturing of a cell that dies from injury. When a cell undergoes apoptosis, it carefully dismantles itself from the inside, packaging its contents into neat little membrane-wrapped bundles that can be safely consumed by macrophages without spilling viral particles everywhere. The cell sacrifices itself to protect the organism. It commits a kind of cellular self-destruction so controlled, so elegant, that it minimizes the collateral damage. The remaining packages can then be processed by macrophages, who will, again, display fragments of the enemy on their surface for other cells to inspect. And there's another arm of the innate system that deserves mention, because it plays a particularly important role in the context of vaccination. Natural killer cells, or NK cells, have one of the more dramatic names in immunology, and they earn it. These cells patrol the body looking for cells that have been infected by viruses or that have become cancerous. They don't need to recognize a specific pathogen the way adaptive immune cells do. Instead, they look for signs of distress. Healthy cells display certain molecules on their surface, essentially identification badges that say, "I'm normal, leave me alone." When a cell is infected by a virus or has become cancerous, those badges are often reduced or absent. NK cells notice the missing badges and kill the cell. It's a simple, elegant system. If you can't prove you're normal, you're destroyed. And there's the complement system, a cascade of roughly 30 proteins that circulate in your blood in an inactive form, like a set of dominoes arranged and waiting. When the first protein in the cascade encounters a pathogen, it activates, and that activation triggers the next protein, and the next, in a rapid chain reaction. The end result is threefold. Some complement proteins coat the surface of the pathogen, making it easier for phagocytes to grab and engulf it, a process called opsonization. Others punch actual physical holes in the membranes of bacteria, killing them directly. And others act as chemical signals that attract more immune cells to the site of infection, amplifying the inflammatory response.
All of these systems, the phagocytes, the NK cells, the complement cascade, the interferons, the process of apoptosis, they form a layered, redundant, overlapping network of defenses that has been refined by hundreds of millions of years of evolution. And they're remarkably effective against the everyday threats you encounter. The vast majority of potentially infectious organisms you come into contact with, and you contact billions every day, are dealt with entirely by these innate systems before you ever feel a symptom. the cough you didn't develop, the infection that never took hold, the cold you didn't catch. Your innate immune system handled it and you never knew. But for the pathogens that have evolved to slip past these defenses, for the ones that can suppress interferons, evade complement, resist phagocytosis, hide inside your own cells, your body needs something more, something that can learn, something that can adapt to threats it has never encountered before. And that something is what your adaptive immune system provides. So your innate immune system is impressive. It's fast, it's ancient, it's always on duty, and it handles the vast majority of threats you encounter every day without you ever knowing about it. Most of the bacteria, most of the fungi, most of the minor viral encounters you have are dealt with entirely by your innate defenses. And the whole thing is over before your adaptive immune system even gets involved. But here's the limitation, and it's a big one. Your innate immune system can't learn. It recognizes the same general patterns it was born recognizing, and it will recognize those same patterns for your entire life. It doesn't get better at fighting a specific pathogen. It doesn't remember previous encounters. If the same bacterium shows up a second time, your innate system responds the same way it did the first time, with the same speed, but also the same limitations. There's no improvement, no specialization, no memory. And for some threats, that's not enough. Some pathogens have evolved to be very good at evading your innate defenses. Viruses that can suppress interferon production. Bacteria that have modified their surface molecules so they don't trigger pattern recognition receptors. Parasites that can hide inside your own cells, cloaked in your own membranes, essentially wearing a disguise that makes them invisible to the first ring of security. Against these threats, your innate system is outmatched. It needs help. And that help comes from the most extraordinary invention in the entire history of biological evolution. A system so sophisticated, so versatile, so breathtakingly clever in its design that immunologists who have studied it for decades still talk about it with something close to awe. Your adaptive immune system. And the thing that makes it remarkable, the thing that separates it from everything your innate defenses can do, is a capability that seems, when you first encounter it, almost impossible. Your adaptive immune system can recognize threats it has never seen before. Threats that have never existed before. Threats that might not exist for another 1,000 years. And it does this using a trick so elegant that when scientists finally figured out how it worked, it won a Nobel Prize. Here's a number that should stop you in your tracks. Your body contains roughly 20,000 genes. 20,000 sets of instructions encoded in DNA that tell your cells how to build the proteins they need to function. 20,000 genes for everything. For the hemoglobin in your red blood cells, for the keratin in your hair, for the collagen in your skin, for every enzyme and receptor and structural protein that keeps you alive. 20,000. And yet, your immune system can produce somewhere in the neighborhood of 1 billion different antibodies, each one shaped slightly differently, each one capable of recognizing a different molecular target. 1 billion different locks, each one waiting for a specific key. How do you get 1 billion different antibodies from 20,000 genes? The math doesn't work. And this paradox, This seemingly impossible discrepancy between the limited number of genes and the staggering diversity of immune responses was one of the great mysteries of 20th century biology. People argued about it for decades, and the answer, when it finally came, was more surprising than anyone had predicted. Here's how it works. And let me use an analogy that I think makes it very intuitive, because the actual molecular biology, while beautiful, is complex enough that the analogy will carry you further tonight. Imagine you have a book, and in this book, there's a page with a long list of nouns separated by commas, hundreds of them. Wolf, there, fox, eagle, serpent, spider, and on and on. Then, on another page, there's a list of adjectives: fierce, swift, cunning, silent, relentless. Hundreds of those too. And on a third page, a list of verbs: hunts, strikes, stalks, waits, devours. More hundreds. Now imagine that you can pick one word from each list and combine them into a phrase. Swift serpent strikes. Fierce eagle hunts. Silent spider waits. The number of possible combinations is enormous. Hundreds times hundreds times hundreds. From 3 modest lists of words, you can generate millions of unique phrases. That's essentially what your immune system does with its genes. The stretches of DNA that encode the business end of an antibody, the part that actually recognizes and grabs onto a target, are not single continuous genes. They're assembled from segments. Variable segments, diversity segments, and joining segments, which scientists call V, D, and J. During the development of each new immune cell, the cell's DNA undergoes a process of deliberate rearrangement. It picks one V segment, one D segment, one J segment, shuffles them together, and the result is a unique combination that encodes a receptor with a unique shape, a shape that will determine what that particular cell can recognize and respond to. This process is called VDJ recombination, and it was worked out primarily by Susumu Tonegawa, who received the Nobel Prize in Physiology or Medicine in 1987 for explaining how the body generates antibody diversity. And the beauty of it is that the recombination is essentially random. Each developing immune cell makes its own random selection from the available segments, and the result is that each cell ends up with a receptor that's different from almost every other cell in the body. You end up with an army of cells, each one tuned to a slightly different target. And here's the part that really matters. Because the combinations are generated randomly, your body doesn't know in advance which pathogens you'll encounter. It can't predict whether you'll be exposed to influenza or tuberculosis or some virus that hasn't even evolved yet. So it takes a different approach entirely. Instead of trying to prepare for specific threats, it prepares for everything. It generates cells with receptors for every conceivable shape, including shapes that correspond to pathogens that don't exist in nature. Shapes that might match a molecule from a virus that won't emerge for another century. Shapes that correspond to nothing at all. Molecules that evolution hasn't produced and perhaps never will. Right now, inside your body, there are immune cells that are waiting for pathogens that don't exist. They have receptors perfectly shaped to grab onto molecular structures that have never appeared on Earth. They will spend their entire lives waiting for a target that will never come. And that's the point. That's the strategy. Because the diversity is so vast, so all-encompassing, that no matter what pathogen shows up, no matter how novel, no matter how unexpected, somewhere in that enormous population of cells, there will be one, maybe a handful, that happens to have just the right receptor shape to recognize it. Not perfectly, maybe, not with exquisite precision, but well enough to grab on, well enough to start the process. Think of it as an army of hunters. millions of them, scattered through your body, each one specialized to hunt a single type of prey. One hunter has spent its entire existence waiting for wolves. Another is equipped to track foxes. Another has been preparing its whole life to hunt a pink unicorn. A creature that doesn't exist.
A mythological animal that no one has ever seen. And the hunter will never find its unicorn. It will live and die without ever encountering the thing it was built to fight. But it's there, ready, just in case. And because your body has so many of these hunters, each waiting for a different target, the odds are overwhelmingly good that when a real pathogen does show up, when something genuinely dangerous slips past your innate defenses and starts causing trouble, one of your hunters will recognize it, because here's what happens next. When one of your hunters finally encounters its target, when a B cell or a T cell with the right receptor shape bumps into the pathogen it was built to recognize, something extraordinary kicks into gear. The cell gets activated, and the first thing it does is start dividing, rapidly, over and over. One cell becomes 2, 2 become 4, 4 become 8, and within days, that single hunter has produced thousands of copies of itself, an entire army of clones, all bearing the same receptor, all tuned to the same target, all hunting the same prey. This is called clonal expansion, and it's one of the most important concepts in immunology. But the process doesn't stop with simple copying. As those clones multiply, they also undergo a process of refinement. Their genes, the genes encoding their receptors, undergo small, targeted mutations. Random variations in the part of the receptor that makes contact with the target. Most of these mutations make the receptor worse, less accurate, a poorer fit. Those cells don't thrive. But some of the mutations make the receptor better, more precise, a tighter grip, a closer match. And those cells, the ones with the improved receptors, get preferential signals to keep dividing. They outcompete their less accurate siblings. It's evolution in miniature. Natural selection playing out inside your body over the course of days rather than millennia.
The end result is a population of cells that are exquisitely tuned to the specific pathogen you're fighting, far more precise than the original hunter that first detected the threat. Immunologists call this process affinity maturation, and it's one of the reasons your immune response gets better over time during an infection. The antibodies your body produces on day 10 are sharper, more targeted, more effective than the ones it produced on day 3. Your immune system is literally learning on the job, getting better at fighting this specific enemy with each passing day. And then, once the infection is cleared, once the pathogen has been eliminated and the battle is won, most of those cloned cells die off. They were built for war, and when the war is over, they disband. But not all of them. A subset of those battle-tested, highly refined cells persist. They settle into quiet corners of your body, your bone marrow, your lymph nodes, and they wait. These are memory cells, and they are the reason you don't get chickenpox twice. They are the reason a disease you survived at age 5 can't touch you at age 70. Because if that same pathogen ever comes back, those memory cells are ready. They don't need days to ramp up. They've already been through the whole process. They remember. And their response, the second time around, is faster, stronger, and more devastating than the first. And there's something else worth knowing about this system. Something that connects the hunters to the innate defenses we talked about earlier in a way that's genuinely beautiful. Remember how I mentioned that macrophages eat pathogens and display fragments on their surface? That process turns out to be absolutely critical for getting the adaptive immune system involved. Because your hunters, your T cells and B cells, don't just wander around bumping into pathogens at random.
Many of them need to be activated by a special class of cells called antigen-presenting cells. And macrophages are one of the most important types. Here's how the handoff works. A macrophage encounters a bacterium or a virus-infected cell. It engulfs the pathogen, digests it, and takes small protein fragments from the pathogen's remains. These fragments, which immunologists call antigens, are then loaded onto special molecules on the macrophage's surface, those MHC molecules I mentioned earlier. The macrophage is essentially wearing the pathogen's clothing, holding up pieces of the enemy's identity like a wanted poster. Now, when a T cell comes along and its randomly generated receptor happens to match one of those displayed fragments, it recognizes the antigen. And that recognition event, that moment when the T cell's receptor locks onto the fragment being held up by the macrophage, is the spark that ignites the adaptive immune response. The T cell activates, it starts dividing, it starts sending chemical signals to other immune cells. It's the moment when the general alarm gets sounded, when the body shifts from the innate response, the non-specific first responders, to the adaptive response, the targeted, precise, remembering system. There's also another type of antigen-presenting cell called a dendritic cell, which is particularly important in the context of vaccines. Dendritic cells are positioned strategically throughout your body, in your skin, in the lining of your nose and lungs and gut, exactly the places where pathogens are most likely to enter. They're like scouts, stationed at the perimeter, constantly sampling their environment, picking up proteins and particles and fragments of whatever is around them. When they encounter something that triggers their pattern recognition receptors, something that looks foreign, they scoop it up, process it, and then migrate to the nearest lymph node. And in that lymph node, they present their findings to the T cells and B cells that are gathered there, waiting. Think about the elegance of this system. You have innate immune cells, the first responders, patrolling the borders. When they find something foreign, they don't just destroy it, they also collect evidence. They gather fragments, and they carry those fragments to the headquarters, the lymph nodes, where the adaptive immune cells are assembled. The innate system tells the adaptive system what to look for. It provides the photograph. This is why vaccines are typically injected into muscle tissue or sometimes into the skin. Those tissues are rich in dendritic cells and macrophages. When the vaccine material arrives, whether it's a weakened pathogen or a killed virus, or a purified protein or a strand of mRNA encoding a viral protein, those antigen-presenting cells pick it up, process it, and carry the fragments to the lymph nodes. And there the long, beautiful process of clonal expansion and affinity maturation begins. Your body has roughly as many bacterial cells living on and inside it as it has human cells of its own. Trillions of them, covering your skin, lining your gut, populating your mouth and nose. Going through the birth canal during birth exposes a newborn to an enormous diversity of these microorganisms. Taking a single swab of your nostril and looking at it under a microscope would reveal a teeming landscape of bacterial life. And your immune system manages all of this constant, overwhelming exposure, every minute of every day. A vaccine, by comparison, is a tiny additional challenge.
One scientist described it as literally a drop in the ocean of what your immune system encounters and manages every single day. Your body produces roughly 1 billion new B and T cells each day just to handle the routine. The additional challenge from a vaccine is vanishingly small in comparison. This is your adaptive immune system. And if you've followed everything so far, you can already see why it explains something that puzzled people for thousands of years. Why, if you survive smallpox, you never get it again. Why a person who recovered from measles in childhood is protected for the rest of their life. The answer is memory cells. Hunters who fought the battle, survived, refined their weapons, and now stand guard permanently. And you can probably also see the outline of a problem. A very serious problem that sits right at the heart of why vaccines exist. Because everything I've just described, the recognition, the clonal expansion, the affinity maturation, the formation of memory, all of it depends on one thing happening first. The pathogen has to get into your body. Your hunter has to encounter its target. And that first encounter, that first meeting between your immune system and a brand new pathogen it has never seen before, takes time. Days. Sometimes a week or more. Your innate system buys you some of that time, slowing the pathogen down, keeping it somewhat in check. But for the really dangerous ones, the ones that have evolved to evade your innate defenses, those days of delay can be the difference between life and death. So the question becomes obvious once you see it. What if you could skip that dangerous first encounter? What if you could give your immune system the information it needed, the photograph of the intruder's face, without exposing it to the actual danger? What if you could trigger the whole beautiful process, the clonal expansion, the affinity maturation, the formation of memory cells, but using something safe? Something that looked enough like the pathogen to train the hunters, but couldn't actually hurt you. What if you could show your body a picture and let it prepare? People stumbled onto a crude version of this idea thousands of years ago. They didn't know why it worked. They didn't know how it worked. They just knew that survivors were safe. And from that single observation, repeated across cultures and centuries, the long, winding road to modern vaccination began. It started with a disease so terrible that entire civilizations organized themselves around the fear of it. And it started with people brave enough, or desperate enough, to try something that sounded completely insane. Smallpox was, for most of human history, one of the most terrifying diseases on the planet. And I want to take a moment to make sure that word, terrifying, lands properly. Because we live in a time when smallpox has been eradicated, when most people have never seen a case, and it's easy to let the horror of it become abstract. So let's make it concrete. The virus that caused smallpox, variola major, killed roughly 30% of the people it infected. 3 out of every 10. And the dying wasn't quick or clean. The disease began with a fever, headaches, body aches, things that might initially feel like a bad flu. But then, after a few days, the rash appeared. Red spots, first on the face and arms, then spreading across the body. The spots became raised bumps, then fluid-filled blisters, then pustules, hard and round and deeply embedded in the skin. In severe cases, the pustules merged together, covering the body in a continuous sheet of swollen, infected tissue. The pain was excruciating. Patients often couldn't eat, couldn't drink, couldn't close their eyes because the pustules covered their eyelids. Death, when it came, usually arrived around the second week, from overwhelming infection or from the body simply giving out. And the survivors, the 7 out of 10 who made it through, were often left permanently scarred, their faces pitted with deep pockmarks where the pustules had been. Many were left blind. In the 18th century alone, smallpox killed an estimated 400 million people. That number is so large, it becomes hard to process. So think of it this way. That's more than the entire current population of the United States, wiped out by a single disease in a single century. But there was something about smallpox that people noticed very early on. Something that, in hindsight, contained the seed of one of the most important medical discoveries in human history. If you survived smallpox, you didn't get it again. Ever. You could nurse smallpox patients, live in a house full of them, and nothing would happen to you. The disease couldn't touch you a second time. This observation was made independently by cultures across the ancient world. They had no concept of viruses. They had no idea what an antibody was, or what a memory cell did, or why the body would develop this mysterious protection. But they could see the pattern with their own eyes. Survival meant safety. And different cultures in different parts of the world responded to this observation in different ways. The most consequential response emerged in China, possibly as early as the 10th century, and it was startlingly direct. Chinese practitioners developed a technique that we now call variolation. The idea was simple, even obvious, once you accepted the premise. If surviving smallpox made you immune, then perhaps the trick was to give someone a mild case of it, a controlled, carefully managed case. Just enough to trigger whatever mysterious protection the body was capable of producing. Their method was to take scab material from a person who had a mild case of smallpox, grind it into a fine powder, and blow it into the nose of a healthy person using a long silver pipe. Sometimes they would use the left nostril for boys and the right for girls, which tells you something about the way that empirical observation and cultural tradition were intertwined in early medicine.
The methods varied from region to region. Some practitioners would take a thread, soak it in the fluid from a fresh smallpox pustule, and draw it through a small scratch on the skin. Others would have the person wear the underclothes of someone who had recently recovered. The methods were crude by modern standards, and the underlying logic was imprecise, but the results were real. Most of the people who were variolated developed a mild case of smallpox, a fever, some pustules, far fewer and far less severe than the natural disease. They were sick for a week or two, and then they recovered. And after they recovered, they were immune. The protection was genuine and lasting. Now, variolation wasn't safe, far from it. About 1 to 2% of the people who underwent the procedure died. They developed full-blown smallpox despite the attempt to give them a mild case, and the disease killed them. 1 to 2%. That sounds terrible. And it was terrible for those individuals and their families. But consider the alternative. The fatality rate from naturally acquired smallpox was 30%. So the math, even without formal statistical methods, was something people could understand intuitively. A 1 in 50 chance of dying from the procedure versus a roughly 1 in 3 chance of dying from the disease. It was a gamble, but for many families, especially during epidemic years, it was a gamble worth taking. And the practice of variolation teaches us something else that's worth sitting with. The decision to variolate your child, to deliberately give them a disease that might kill them, in order to protect them from a disease that would almost certainly kill them, was one of the most agonizing decisions a parent could face. And yet, millions of families across dozens of cultures, across centuries, independently came to the same conclusion. The risk was worth taking.
The small danger of the procedure was preferable to the large danger of the disease. This calculation, this weighing of a known small risk against a larger but uncertain one, is the fundamental logic underlying all of vaccination. And it's a calculation that makes many people deeply uncomfortable, because it acknowledges something we'd rather not think about: that perfect safety doesn't exist. That every medical intervention, every medicine, every procedure carries some degree of risk. And that the relevant question is never, is this perfectly safe? Because nothing is. The relevant question is always, is this safer than the alternative? And the alternative to vaccination, for most of human history, was the disease itself. The numbers tell the story more clearly than any argument. Variolation, 1 to 2% fatality. Natural smallpox, 30% fatality. The gap between those numbers is the space in which all of vaccinology lives.
Every vaccine ever developed has been an attempt to make that gap as large as possible, to push the risk of the intervention down as close to zero as the science allows, while maintaining enough immunological activity to provide genuine protection. And over 2 centuries, the gap has gotten wider and wider. Modern vaccines have side effect profiles that would have seemed miraculous to the practitioners who blew scab powder into children's nostrils in 10th century China. But the fundamental logic hasn't changed. You're still weighing one risk against another. You're still making a bet. The bet has just gotten dramatically safer. And the people who refuse to make that bet, who look at the 1 in a million risk of a serious vaccine side effect and decide it's not worth it, are often making a mistake that's easy to understand, but hard to forgive. They're comparing the risk of the vaccine not to the risk of the disease, but to the risk of doing nothing. And doing nothing feels safe. It feels like the neutral option, the default, the path of least resistance. But doing nothing is also a choice, and it carries its own risks. The risk of contracting the disease, the risk of spreading it to others who are more vulnerable, the risk of being the reason an outbreak starts in a community that had been protected. During the height of the COVID pandemic, unvaccinated individuals were 12 times more likely to be hospitalized than vaccinated individuals. 12 times. That gap, that enormous disparity in outcomes, is the modern version of the variolation math. The vaccine isn't perfect. It has side effects, most of them minor, a few of them serious, all of them rare. But the disease is worse. The disease has always been worse. And the people who understood that, from the Chinese practitioners of the 10th century to the parents who lined their children up for Salk's polio vaccine in 1955, were making the same essential calculation. The small known risk of the intervention is better than the large, uncertain risk of the disease. Variolation spread along trade routes. By the 17th century, variants of the practice were being used across parts of Asia, Africa, and the Ottoman Empire. And this is where one of those wonderful human stories enters the picture. Her name was Lady Mary Wortley Montagu. She was a British aristocrat, a writer, and by all accounts genuinely formidable. In 1715, smallpox nearly killed her. She was 26 years old, considered one of the great beauties of London society, and the disease ravaged her face. She survived, but the scars were deep and permanent. Her famous beauty was gone. Then, 2 years later, her husband was appointed ambassador to the Ottoman Empire, and she travelled with him to Constantinople. And there, in the homes of Ottoman women who befriended her, she witnessed something remarkable. Variolation being practiced openly, routinely, and successfully. The Ottoman women showed her how it was done. An old woman would come with a nutshell full of smallpox matter, scratch it into a vein in the arm, and within 8 days, the patient would have a mild fever and a handful of small spots. Then recovery, then immunity. Lady Mary was captivated. She had her own son variolated right there in Constantinople, supervised by the embassy surgeon. The boy came through it fine. When she returned to England, she became a fierce advocate for the practice. She had her daughter variolated publicly in London in 1721 during a smallpox epidemic as a deliberate demonstration. And she pushed hard to get the British medical establishment to pay attention. The establishment pushed back, and the reasons are worth examining because the same pattern would repeat itself throughout the entire history of vaccines. Part of it was legitimate scientific caution. The procedure was dangerous after all. Part of it was professional jealousy. Who was this aristocratic woman to tell physicians their business? Part of it was cultural prejudice. This was a technique from the Ottoman Empire, from Turkish women, and some English doctors couldn't accept that foreigners, and women at that, might know something they didn't. And part of it was religious objection. Several prominent clergymen argued that smallpox was God's will, and deliberately interfering with it was sinful. But the evidence kept accumulating. King George I authorized a trial on condemned prisoners at Newgate Prison. The prisoners survived and proved immune when later exposed to smallpox. Then the procedure was tested on orphan children, because apparently in the 18th century, prisoners and orphans were considered acceptable experimental subjects. The children survived. Eventually, members of the royal family were variolated, and the practice spread. But the fundamental problem was always there. You were still giving people real smallpox, a milder form usually, but real smallpox. And that 1 to 2% fatality rate meant that for every 100 people you variolated, 1 or 2 might die, and the variolated could spread the disease to others. What was needed was something better, something safer, and that something was about to come from the most unlikely source. Not from a great university, not from a royal physician, but from a country doctor who listened to what milkmaids told him about their hands. Edward Jenner was born in 1749 in Berkeley, a small market town in Gloucestershire, in the green, rolling dairy country of Western England. He was the son of a clergyman, orphaned young, raised by an older brother. As a boy, he was variolated against smallpox, and by his own account, it was a miserable experience. Weeks of preparation, involving purging, fasting, and bleeding before the actual procedure. It left enough of an impression that he would remember it for the rest of his life. He became a country doctor, a general practitioner, treating farmers and their families. Not a glamorous career. He spent his days riding between farms on horseback, setting broken bones, delivering babies, treating fevers. But he was observant, curious, and he had the kind of patient, methodical mind that notices things other people overlook.
and there was something he'd been hearing from the milkmaids of Gloucestershire that had been nagging at him for years. Milkmaids who worked closely with cows sometimes caught a disease called cowpox. It was an occupational hazard. Cowpox produced sores on the cow's udders, and the milkmaid's hands, in constant contact with those udders during milking, would develop similar sores. Blisters on the fingers and palms, sometimes a mild fever, a few days of discomfort, and then recovery. Compared to smallpox, cowpox was nothing, a nuisance. But here's what the milkmaids said, and what local farmers confirmed.
People who'd had cowpox didn't get smallpox.
The milkmaids, with their rough, blistered hands, seemed to be immune. It wasn't a secret. Everybody in dairy country knew it. Nobody in London paid attention to it. Jenner wasn't even the first to act on this knowledge. A farmer named Benjamin Jesty had deliberately infected his wife and 2 sons with cowpox back in 1774, using a darning needle to scratch material from a cow's sore into their arms. His wife had a bad reaction, but recovered, and the family remained immune to smallpox for the rest of their lives. But Jesty was a farmer. He didn't write anything down. He didn't publish a paper. His contribution was largely forgotten. What Jenner did was test the folk observation methodically. On May 14, 1796, he took material from a cowpox sore on the hand of a young milkmaid named Sarah Nelms.
Sarah had caught cowpox from a cow named Blossom, which is one of those details that feels almost too pastoral to be real, but apparently is. Jenner scratched the cowpox material into the arm of an 8-year-old boy named James Phipps, the son of his gardener. The boy developed a mild cowpox infection. A few sores, a slight fever, nothing serious. He recovered within days. And then, about 6 weeks later, Jenner did something that would make any modern ethics committee collapse. He variolated the boy with actual smallpox. He deliberately exposed this child to one of the deadliest diseases in human history, betting everything on the hunch that cowpox would protect him. If Jenner was wrong, James Phipps could have developed smallpox and died. Jenner's career, his reputation, his freedom, all of it hung on the outcome. And it worked. James Phipps showed no sign of smallpox. Jenner exposed him again later, and again, nothing. The boy was immune.
Think about what Jenner understood and what he didn't. He had no idea what a virus was. He had no microscope powerful enough to see a viral particle. He didn't know about antibodies or T cells or any of the molecular machinery we've been talking about. He was operating on pure observation and a willingness to take a terrifying risk with someone else's child. And yet, the principle he demonstrated, that exposure to a related but less dangerous pathogen could confer protection against a deadly one, is the foundation of everything that followed. Jenner wrote up his findings and submitted them to the Royal Society. They rejected the paper. They told him he needed more evidence, so he gathered more cases and in 1798 published the results himself. In the pamphlet, he coined the term vaccine, from the Latin word vacca, meaning cow. The reaction was, predictably, a mess. Some doctors embraced it immediately, others attacked it furiously.
There were satirical cartoons showing vaccinated people sprouting cow parts from their bodies. The anti-vaccination movement existed from literally the very first vaccine. And to be fair, some of the early concerns weren't unreasonable. The cowpox material wasn't standardized. Sometimes it was contaminated, sometimes it didn't work. And the specific fears have shifted over the decades, morphing to fit the cultural anxieties of each era. But the underlying pattern remains remarkably consistent. In 1798, people feared that the vaccine would turn them into cows. In the 21st century, a different fear took hold, one that would prove far more damaging and far more persistent. In 1998, a British doctor named Andrew Wakefield published a paper in The Lancet, one of the world's most respected medical journals, suggesting a possible link between the MMR vaccine and autism. The paper was based on 12 children, 12. A sample so small that no serious statistical conclusion could be drawn from it. But the paper was published in a prestigious journal, and it received enormous media attention, and the damage was swift and devastating. Investigation after investigation found problems with Wakefield's work. Some of the children in his study had shown signs of developmental issues before they received the MMR vaccine, but this was not disclosed. Some diagnoses had been altered. The timing of symptom onset had been misrepresented, and it turned out that Wakefield had undisclosed financial conflicts of interest. He held a patent on a competing single-component measles vaccine. He had received funding from lawyers who were suing vaccine manufacturers. He wasn't trying to protect children from vaccines. He was trying to discredit one vaccine to promote his own. The Lancet retracted the paper. Wakefield was stripped of his medical license. And in the years that followed, study after study, involving millions of children, found no connection whatsoever between the MMR vaccine and autism. A Danish study of over 600,000 children, a meta-analysis of 138 studies covering more than 23 million children. The evidence is as overwhelming as scientific evidence can be. Vaccines do not cause autism, and yet the myth persists. It persists because it was planted at a moment of parental vulnerability, when parents of children with autism were desperate for answers and were offered a villain. It persists because the emotional power of a personal story, "My child was vaccinated and then developed autism," is almost impossible to counter with statistical arguments, even when the statistics are unambiguous. It persists because the retraction of a scientific paper receives a fraction of the attention that its original publication received.
And it persists because there are people, Wakefield prominent among them, who have built careers and audiences and financial empires on promoting the myth. The real tragedy is not just the myth itself, but its consequences. Measles vaccination rates dropped in the UK and in parts of the United States. Measles outbreaks followed. Children died. Of measles, a disease that we had the tools to prevent, tools that had been proven safe and effective over decades, in hundreds of millions of doses. Children died because a fraudulent paper in a medical journal convinced their parents not to vaccinate them. It is, as one scientist described it, one of the most shameful episodes in the history of public health communication. But the results spoke. Vaccinations spread across Europe, then to the Americas, then worldwide. Napoleon had his army vaccinated. Thomas Jefferson promoted it in the United States. And the Spanish Crown organized one of the most remarkable public health missions in history. A story that deserves a moment because it shows the extraordinary lengths people went to in an age before refrigeration. In 1803, King Charles IV of Spain commissioned the Royal Philanthropic Vaccine Expedition, led by a physician named Francisco Xavier de Balmas. The goal was to bring vaccination to the Spanish colonies. But the cowpox vaccine material was alive. It was a living virus, and it needed living tissue to stay viable. You couldn't just bottle it and ship it across an ocean. Balmas's solution was extraordinary, and by modern standards, deeply troubling. He brought 22 orphan boys onto the ship, young children who had never had cowpox or smallpox. During the voyage,
He would vaccinate 2 boys at a time, wait for their cowpox sores to develop, and then use material from those sores to vaccinate the next 2. A living chain of infection, arm to arm, child to child, carried across the Atlantic and eventually the Pacific. The timing had to be precise. The sores needed to be at exactly the right stage when the next pair was vaccinated. If the chain broke, the expedition would fail. Balmis managed it. The vaccine reached Mexico, Colombia, Venezuela, Peru, the Philippines, and China. It saved untold thousands of lives. It also used children as biological shipping containers. History is rarely as clean as we'd like it to be. There's a detail about the broader spread of vaccination in the decades after Jenner that's worth knowing, because it illuminates how scientific ideas travel and how they're received in different cultural contexts. Vaccination reached India through British colonial channels in the early 1800s, and it was met with a complex mix of acceptance and resistance that had as much to do with politics as with medicine. Some Indian communities embraced it quickly, others rejected it, not because they doubted its efficacy, but because they distrusted the colonial authorities promoting it. They had their own variolation traditions, practiced for centuries, and they didn't appreciate being told by foreign rulers that their methods were inferior. This pattern, where resistance to vaccination is driven less by medical skepticism than by social and political distrust, repeats throughout history and persists today. People don't refuse vaccines because they've carefully evaluated the scientific literature and found it wanting. They refuse because they don't trust the institutions promoting them, because they feel talked down to, or manipulated, or left out of the decision-making process, because they've been burned before by authorities who told them something was safe and turned out to be wrong. The Qatar incident, thalidomide, the Tuskegee syphilis study. These betrayals of trust, some involving vaccines and some not, cast long shadows.
And you can't address vaccine hesitancy purely with scientific arguments, because the hesitancy often isn't about the science. It's about power and trust and who gets to make decisions about your body. The most effective vaccination campaigns in history have understood this. They've worked with local communities rather than imposing solutions from above. They've trained local healthcare workers to administer vaccines rather than sending in outsiders. They've explained the risks honestly rather than dismissing concerns. And they've shown respect for people's autonomy, even while strongly encouraging them to get vaccinated. Vaccination works best when it's offered, not ordered, when it's explained, not marketed, when the people promoting it are willing to say, here's what we know, here's what we don't know, and here's why we think the benefits outweigh the risks, rather than simply demanding compliance. And here's the thing about the 80 years after Jenner.
For roughly 8 decades, vaccination worked brilliantly. Wherever it was practiced consistently, smallpox rates plummeted. The evidence was overwhelming, and yet nobody understood why. The immune system as a concept didn't exist. Germ theory was decades away. Jenner's vaccine was a tool that worked magnificently in practice, while being almost entirely opaque in theory. The doing came first. The understanding followed. And the person who would begin to bridge that gap, who would take what Jenner had done with cowpox and turn it into a general principle applicable to any disease, was not a doctor at all. He was a French chemist who had started his career studying crystals and sour wine. And the path he took from fermentation to immunization is one of those winding, unpredictable journeys that reminds you the biggest breakthroughs often come from the most unexpected directions.
Louis Pasteur was born in 1822 in Dole,
a small town in eastern France, the son of a tanner who had served in Napoleon's army. The family wasn't wealthy, but they valued education. The young Louis had a stubborn streak and an almost obsessive attention to detail that his teachers noticed early on. His early work had absolutely nothing to do with medicine. He was studying crystals, tartaric acid crystals, the tiny formations that appear in wine barrels during fermentation. He noticed that these crystals came in two forms that were mirror images of each other, like left and right hands. Identical in chemical composition, but different in three-dimensional structure. This was a foundational discovery in what we now call stereochemistry, and Pasteur figured it out in his 20s, separating crystals by hand with tweezers under a microscope. But it's worth spending another moment with Pasteur's path from crystals to medicine, because it illustrates something important about how scientific breakthroughs actually happen. They rarely follow a straight line. Pasteur didn't wake up one morning and decide to develop vaccines. He started with crystals. Crystals led him to fermentation. Fermentation led him to microorganisms. Microorganisms led him to germ theory. And germ theory led him to ask whether you could harness microorganisms to prevent disease. Each step followed logically from the last, but the overall journey, from tartaric acid in wine barrels to a 9-year-old boy surviving rabies, would have been impossible to predict at the outset. This is how discovery usually works. You follow the question in front of you, and it leads you somewhere you didn't expect. Katalin Karikó wasn't trying to develop a COVID vaccine when she started working on mRNA in the 1980s. COVID didn't exist yet. She was trying to understand a fundamental biological mechanism. And that fundamental understanding, built up over decades of work that most of the world ignored, turned out to be exactly what was needed when a pandemic struck 30 years later. The lesson, if there is one, is that basic science matters, even when it seems disconnected from practical applications. The copying machine where Karikó met Weissman, the tangerine tree whose thorn Metchnikoff stuck into a starfish larva, the wine barrels whose crystals captured Pasteur's attention. These seemingly small, seemingly unrelated moments turned out to be the first steps on paths that would save millions of lives. And you can never tell at the beginning which paths those will be. That crystal work led him to study fermentation itself. The French wine and beer industries were having problems, batches going sour, spoiling unpredictably. Pasteur was asked to investigate, and what he found overturned centuries of assumption.
He demonstrated that fermentation wasn't just a chemical process, it was biological. Living microorganisms, yeasts, were converting sugar into alcohol. And when the wrong microorganisms got in, you got vinegar instead of wine, or sour beer instead of good beer. This was radical because the dominant theory of the time was spontaneous generation, the idea that living things could arise from non-living matter, maggots on rotting meat, mold on bread. The prevailing explanation was that these organisms simply materialized out of nothing. Pasteur demolished it. His most famous experiment used specially designed flasks with long, curved, swan-like necks. He boiled broth to sterilize it, then left the flasks open. The curved neck allowed air in, but trapped dust and microorganisms in the bend. The broth stayed clear. For months, for years, nothing grew. But the moment he broke the neck, or tilted the flask so the broth touched the contaminated bend, within days, it was cloudy with microbial life. The air itself wasn't generating organisms. The microorganisms were already there, floating invisibly, waiting to land on something they could grow in. And here's where the thread connects. If microorganisms caused fermentation, could they also cause disease? This was the intellectual leap that bridged Pasteur's work on wine with what would become his work on vaccines. Robert Koch in Germany was asking similar questions, identifying specific bacteria responsible for specific diseases. The two men didn't get along. There was a genuine Franco-German rivalry sharpened by the politics of the era. But together, often in bitter competition, they built the framework of germ theory. Diseases were caused by specific microorganisms. Pasteur started thinking about Jenner's vaccine, and he saw something important. Jenner's cowpox vaccine had been a gift from nature. Cowpox just happened to be similar enough to smallpox to provide cross-protection. Nobody had engineered that. What if you could do it deliberately? What if you could take a dangerous pathogen, weaken it, and turn it into a vaccine? The breakthrough came in 1879. Pasteur's laboratory had been working with chicken cholera, a devastating disease that killed poultry rapidly. They had bacterial cultures growing in the lab, and then Pasteur's assistant, Charles Chamberlain, went on vacation. The cultures were left sitting on the bench, exposed to air, neglected for weeks. When Chamberlain returned and injected chickens with these old cultures, the chickens got mildly sick but didn't die. They recovered. A lesser scientist might have thrown the degraded cultures away, but Pasteur took those same chickens and injected them with fresh, full-strength, lethal chicken cholera bacteria. Every unvaccinated chicken died.
The vaccinated chickens survived. The bacteria, left sitting exposed to oxygen, had lost some of their virulence. They'd been attenuated, weakened by the conditions, and this weakened form could still stimulate the immune system, could still show the body enough of the pathogen for it to learn and remember, but couldn't cause the full disease. Pasteur had manufactured the photograph. He hadn't found a naturally occurring cousin of the pathogen the way Jenner had. He had taken the pathogen itself and weakened it. Pasteur, who respected Jenner deeply, deliberately used the word vaccine to describe his creation, even though it had nothing to do with cows. Vaccination was no longer limited to one disease. It was a strategy, a general method. He moved fast. After chicken cholera came anthrax, a disease devastating livestock across Europe.
In 1881, at a farm called Pouilly-le-Fort, in front of journalists, politicians and skeptics, Pasteur vaccinated 25 sheep with his weakened anthrax preparation. 25 other sheep were left unvaccinated. Then all 50 were injected with lethal anthrax. The results were devastating in their clarity. Every vaccinated sheep survived. Every unvaccinated sheep died or was dying. I should mention that Pasteur's private notebooks, opened decades after his death, revealed that the vaccine he used may not have been prepared exactly the way he publicly described. There's evidence he used a chemical method developed by a rival, Henri Toussaint, rather than the oxygen attenuation technique he claimed. Pasteur was brilliant but fiercely competitive and concerned with priority. The science worked. The results were real. But the backstory was messier than the public version. The story of science usually is. After anthrax came rabies, and rabies was different because the pathogen was invisible. Pasteur couldn't see it under his microscope, couldn't grow it in a dish. We know now that rabies is caused by a virus, but the concept of a virus as we understand it didn't exist yet. Pasteur called the invisible agent a virus, using the old Latin word for poison, but couldn't characterize it further. He worked with infected tissue. He took spinal cord material from rabid rabbits and weakened it by drying it over potassium hydroxide. The longer the drying, the weaker the preparation. Start with the weakest and work up. gradually building immunity. The real test came in July 1885, when a 9-year-old boy named Joseph Meister was brought to Pasteur. The boy had been bitten 14 times by a rabid dog. Without treatment, rabies was essentially a death sentence. Once symptoms appeared, the fatality rate was 100%, and the death was horrible.
Neurological deterioration, hydrophobia, delirium, paralysis. Pasteur agonized. He wasn't a physician. He'd never tested the treatment on a human. If it failed, or if it caused rabies, he could be charged with murder. He consulted doctors. He weighed the impossible math. The boy was almost certainly going to die without treatment. Pasteur decided to proceed. Over 11 days, Joseph Meister received 13 injections of increasingly potent rabies material. And then, everyone waited. Rabies has a long incubation period, weeks to months. Every day without symptoms was a small victory. The days passed, the weeks passed. The symptoms never came. Joseph Meister survived. He grew up, lived to adulthood, and eventually became the gatekeeper at the Pasteur Institute in Paris. The news electrified the world. And the treatment worked, again and again, on people who traveled to Paris from across the globe. Not every time. Some who arrived too late, after the virus had reached the brain, still died. But the success rate was remarkable enough to be undeniable. Pasteur's rabies treatment also revealed something interesting about the way vaccination can work that's worth pausing on, because it shows up again in modern medicine. Most vaccines are given before exposure. You vaccinate a child against measles before they've ever encountered the measles virus. The whole point is to build immunity in advance, so the body is ready when the pathogen arrives. But Pasteur's rabies treatment was given after the bite. After the patient was already potentially infected, the reason this could work is specific to rabies. The rabies virus enters through the wound and then travels along the peripheral nerves toward the brain. And it travels slowly. The journey from the bite site to the central nervous system can take weeks, sometimes months. That slow travel provides a window. If you can inject vaccine material and provoke an immune response fast enough, the body can produce antibodies and T cells that neutralize the virus before it reaches the brain. It's a race, and with rabies, the vaccine usually wins because the virus takes so long to reach its destination. This distinction between preventive vaccination and post-exposure prophylaxis is still used today. If you get bitten by a potentially rabid animal in the 21st century, you'll receive a series of rabies shots that are conceptually descended from what Pasteur developed, though the modern versions are far safer. It's one of the few situations where vaccination after exposure is standard practice. And there's a poignant footnote to the Joseph Meister story. Meister grew up, became the gatekeeper of the Pasteur Institute, and lived a quiet life in Paris. When the Nazi forces occupied France in 1940, German soldiers demanded that Meister open Pasteur's burial crypt, which was in the Institute building. Meister, rather than comply, took his own life. He was 64 years old. It's one of those details that reminds you that the history of science, for all its abstractions and molecular mechanisms, is ultimately a history of people. People with fears and loyalties and breaking points. By the late 1880s, the landscape of vaccine science had been transformed. Jenner had shown that vaccination was possible. Pasteur had shown it was a general principle. But through all of this, through decades of increasingly successful vaccination, there was still that fundamental question. What was actually happening inside the body? When a vaccine did its work, what was the mechanism? The answers were about to start emerging from two completely different directions. And the debate between these two camps would rage for 20 years before anyone realized that both sides had been right all along. The person who opened the first window into understanding the immune system at the cellular level was a zoologist named Elie Metchnikoff, and the circumstances of his discovery were almost comically unlikely. It was 1882. Metchnikoff was at a marine biology station in Messina on the coast of Sicily. His family had gone out for the evening to the circus. He was alone in the laboratory, working late. He had been studying starfish larvae, tiny, transparent creatures that you could see right through under a microscope. And on this particular evening, he did something simple. He took a small thorn from a tangerine tree in the garden outside the lab and slipped it into the body of one of the larvae. And then he watched. Over the next several hours, tiny mobile cells within the larva began to gather around the thorn.
They moved toward it, clustering against it, pressing up against the foreign object. By morning, the thorn was coated with them. These cells were trying to engulf it, trying to eat it. Here was a creature with no blood vessels, no lymph nodes, no antibodies, no adaptive immune system of any kind. A starfish larva is about as simple an animal as you can study. And yet, it had cells that recognized something foreign and moved toward it with purpose. Metchnikoff realized he was looking at something fundamental, something ancient, something that probably existed in all animals, from the simplest to the most complex. He called these cells phagocytes, from the Greek words for eating and cell, and he proposed a radical theory. The primary defense of living organisms against infection was not some chemical property of the blood. It was cellular. Living cells that roamed the body, detected invaders, and devoured them. This was deeply controversial, because at exactly the same moment, researchers in Germany were pursuing a completely different explanation. They were finding that blood serum, the liquid part of blood with all the cells removed, could neutralize bacterial toxins and kill bacteria entirely on its own. No cells required, just the fluid. The key figures in this opposing camp were Emil von Behring and Kitasato Shibasaburo, who in 1890 published a landmark paper showing that the serum of animals immunized against diphtheria or tetanus could protect other animals from those diseases. They called the protective substances antitoxins. We'd call them antibodies today. And von Behring turned the discovery into something immediately, urgently practical. Diphtheria was a horrifying childhood disease. It killed tens of thousands of children every year in Europe and America.
The bacterium produces a powerful toxin that damages the heart and nervous system, and it creates a thick, leathery membrane in the throat that can literally suffocate a child. Parents called it the strangling angel of children. Behring showed that you could immunize a horse against diphtheria by giving it increasing doses of the toxin until the horse's blood was rich with antitoxin. You could then extract the horse's serum and inject it into a sick child. The borrowed antitoxin would neutralize the diphtheria toxin in the child's body. Children who would have died were saved. This was passive immunization. Behring was giving children someone else's antibodies, borrowed weapons, rather than training the children's own immune systems. The protection was immediate but temporary, because the borrowed antibodies eventually broke down, and the child's own system had never learned to make them.
But in the midst of a diphtheria epidemic, temporary protection was better than no protection at all. Behring was awarded the very first Nobel Prize in Physiology or Medicine in 1901. So picture the scientific landscape. You've got Mechnikov in Paris, arguing that immunity is fundamentally about cells eating pathogens. And you've got the German school arguing that immunity is fundamentally about soluble chemicals in the blood neutralizing toxins. Cells versus serum. Eaters versus chemicals. And for about 20 years, this was one of the most heated debates in all of biology. And here's how it resolved. They were both right. Each side was looking at a different arm of what turned out to be an enormously complex system. The immune system doesn't choose between cellular defense and chemical defense. It uses both, simultaneously, in carefully coordinated layers. Mechnikov's phagocytes, the macrophages and neutrophils we've already talked about, are a crucial part of innate immunity. And the antibodies that Behring and Kitasato discovered are the signature weapon of adaptive immunity. The fast, non-specific response and the slow, specific response working together. Neither one, alone, tells the whole story. Mechnikov had to wait until 1908 for his Nobel, which he shared with Paul Ehrlich, who had helped bridge the gap between the two camps. The Nobel Committee, in its way, was acknowledging that both perspectives were essential. And Behring's diphtheria antitoxin, as life-saving as it was, also taught an early and painful lesson. Some patients who received the horse serum developed a reaction, not to the diphtheria toxin, but to the horse proteins themselves. Their immune systems recognized the foreign horse serum as an invader and attacked it, causing fever, rashes, joint pain, sometimes worse. This was called serum sickness, and it was one of the first clear demonstrations that the immune system doesn't always hit the right target. Sometimes it overreacts. Sometimes it attacks things it shouldn't. Managing those misfires would become one of the central challenges of vaccine development for the next century and beyond. And this distinction between passive and active immunity, first demonstrated by Behring's work with diphtheria antitoxin, is one we still use every day in medicine. When you receive a tetanus shot after stepping on a rusty nail, you might receive both things at once: a vaccine booster, to re-stimulate your own immune memory and get your body producing fresh antibodies, and a dose of tetanus immunoglobulin, which is preformed antibodies harvested from donated blood. The immunoglobulin gives you immediate passive protection right now, while the vaccine activates your own immune system to ramp up its own response over the coming days and weeks. Two strategies, passive and active, working in tandem. Same basic logic that Behring pioneered over 100 years ago. The serum sickness problem that plagued Behring's horse-derived antitoxin also pointed towards something that would become increasingly important as vaccine science progressed. The immune system is powerful, and that power cuts both ways. It can protect you, but it can also, in certain circumstances, overreact. An excessively strong immune response to a vaccine or to the vehicle delivering the vaccine can cause side effects. Fever, swelling at the injection site, fatigue. These are all signs that the immune system is doing exactly what it's supposed to do, recognizing something foreign and mounting a response. They're uncomfortable, but they're evidence that the vaccine is working. In very rare cases, the immune response can be more serious.
Anaphylaxis, a severe, whole-body allergic reaction, occurs in roughly 1 case per million injections, according to large surveillance studies. That's an extraordinarily low rate, but it's real, which is why people are typically asked to wait for 15 to 30 minutes after receiving a vaccine, so that if anaphylaxis does occur, it can be treated immediately with epinephrine. The monitoring system is built into the process, precisely because the history of vaccinology, from serum sickness to the Cutter incident, has taught that even the safest medical interventions carry some small degree of risk. And the way to manage that risk is not to pretend it doesn't exist, but to build systems that detect and respond to it rapidly. There's a concept in medicine
that a doctor who helped design one of the most important modern vaccines described very simply. The medical definition of safety, he said, is that the benefits have to clearly and definitively outweigh the risks.
Not that there are no risks. There are always risks. The question is whether the protection you gain from the vaccine is large enough and important enough to justify the small, known, and manageable risks that come with it. And for the vast majority of vaccines on the recommended schedule, the answer is overwhelmingly yes. The diseases they prevent are far more dangerous than any documented side effect of the vaccines themselves. This isn't a matter of opinion. It's a conclusion drawn from clinical trials involving tens of thousands of participants and post-licensure surveillance data involving hundreds of millions of doses. But while the debate about cells and chemicals was playing out in laboratories across Europe, there was another challenge emerging that would test everything they'd learned. There was a whole category of pathogen that was proving far harder to work with than bacteria. A category so small, so elusive, so fundamentally strange that it took decades just to prove these things existed. By the late 1800s, the toolkit for working with bacteria was getting better every year. Scientists could grow them in culture dishes, study them under microscopes, identify which species caused which diseases. But some diseases stubbornly refused to yield to these methods. The infectious agent couldn't be seen under the best available microscopes. It couldn't be grown on agar plates. And most puzzlingly, it could pass through filters with pores fine enough to trap every known bacterium. Whatever was causing these diseases was smaller than anything scientists had ever encountered. In 1892, a Russian botanist named Dmitry Ivanovsky found that the agent causing tobacco mosaic disease passed right through porcelain filters that should have stopped anything cellular. He reported the finding, but didn't fully appreciate what it meant. A few years later, the Dutch microbiologist Martinus Beijerinck repeated the work and drove the point home. This was not a bacterium. It was something fundamentally different, something that could only reproduce inside living cells. Beijerinck called it a contagium vivum fluidum, a contagious living fluid. And for decades after Beijerinck's work, viruses remained almost ghostly. Scientists knew they existed because they could see their effects, the diseases they caused, the cells they destroyed. But they couldn't see the viruses themselves. The technology simply wasn't available. Electron microscopy, which would finally allow researchers to visualize viral particles directly, didn't arrive until the late 1930s and early 1940s. When scientists finally saw what viruses looked like, the images were startling. These weren't cells. They weren't really organisms at all, in the conventional sense. They were geometric structures, crystalline, almost architectural. Icosahedral shells of protein surrounding tightly packed nucleic acid. They looked more like engineering blueprints than living creatures. And in a sense, that's what they are. A virus is, at its most fundamental, a set of instructions wrapped in a protective coat. It has no metabolism. It can't produce energy. It can't reproduce on its own. It's entirely dependent on the cellular machinery of its host. It hijacks the cell's ribosomes, its energy supply, its protein-making factories, and repurposes them all to produce copies of itself. Some biologists argue about whether viruses should even be considered alive. They occupy a strange twilight zone between chemistry and biology, between the living and the non-living. And this peculiar nature is what made them so challenging for vaccine developers. With bacteria, you had a free living organism that you could grow in broth, manipulate, weaken, kill. With viruses, you had something that only came to life, if you could call it that, inside another living cell. You couldn't grow them independently. You needed host cells. living tissue. And providing that living tissue at the scale needed for vaccine manufacturing was one of the great practical challenges of 20th century biology. The solution came in stages. First, live animals. Jenner's cowpox vaccine was essentially grown in cows, or rather, in the natural infection cycle of cowpox in cows, and then transferred to humans. Pasteur grew rabies virus in the spinal cords of rabbits. These methods worked, but they were slow, difficult to standardize, and carried the constant risk of contamination with other infectious agents that the animals might be harboring. Then came eggs. The discovery that certain viruses could be grown in the developing embryos of fertilized chicken eggs was a major advance.
Max Tyler used this technique to develop the yellow fever vaccine in the 1930s, work that earned him the Nobel Prize in 1951. The egg-based method proved remarkably versatile. By the middle of the 20th century, influenza vaccines, yellow fever vaccines, and several others were all being produced in eggs. If you've ever been asked before getting a flu shot whether you're allergic to eggs, this is why. The technique is so well established that a significant portion of the world's flu vaccine supply is still produced this way. Billions of eggs every year. Chickens raised in ultra-clean environments, their eggs destined not for breakfast, but for public health. And then came tissue culture, Ender's breakthrough that we've already discussed, which opened the door to growing viruses in cells in a dish rather than in a whole organism. This was the advance that made the polio vaccines possible, and that more broadly transformed virology from a largely observational science into an experimental one. Each of these advances in growing and handling viruses corresponded to a new generation of vaccines, and each generation brought the photograph into sharper focus while reducing the risk. By the early 1950s, with Ender's tissue culture technique making large-scale virus production practical for the first time, everything was in place for an assault on the most feared disease in America. Polio struck mostly in summer, mostly in children. Most infections were mild, barely noticeable, but a fraction of cases resulted in the virus attacking motor neurons in the spinal cord, the nerve cells that control voluntary muscle movement. When those neurons were destroyed, the muscles they controlled were paralyzed permanently. Children woke up unable to move their legs, unable to stand. In the most severe cases, unable to breathe. The images from that era are haunting. Hospital wards filled with rows of iron lungs, large metal cylinders that encased a patient's body and used air pressure changes to inflate and deflate the chest, doing the work that paralyzed muscles could no longer do. Children's faces poking out from the open end. Public swimming pools closed every summer. Parents kept children indoors. The dread was pervasive. Franklin Roosevelt had been paralyzed by what was almost certainly polio in 1921. His involvement helped create the National Foundation for Infantile Paralysis, later known as the March of Dimes, which funded polio research on an unprecedented scale through small donations from ordinary Americans. Dimes and quarters, millions of them, mailed in by families desperate for a solution. Parents lived in dread every summer. Public pools were drained and roped off. Birthday parties were cancelled. Playgrounds stood empty. Mothers washed their children's hands obsessively and watched for the first sign of a fever with a fear that bordered on panic. Newspapers ran daily counts of new cases the way they would later run stock market numbers. The March of Dimes, with its genius for grassroots fundraising,
sent collection cards to every household in America, and families who had barely enough to feed themselves dropped their dimes and quarters into the slots, because this was a disease that didn't discriminate. It struck rich neighborhoods and poor ones. It struck healthy, well-nourished children as easily as malnourished ones. It seemed almost random in its cruelty, which made it even more terrifying. You couldn't protect your child by keeping the house clean, or feeding them well, or praying hard enough. Polio came for whoever it wanted, and there was nothing you could do about it except hope.
The National Foundation for Infantile Paralysis, with Roosevelt's personal involvement and later his legacy, channeled that collective fear into collective action on a scale that American public health had never seen. They funded research laboratories, they paid for iron lungs, they organized rehabilitation programs, they subsidized hospital care for polio patients, and eventually they funded the vaccine trials that would change everything. The money came from ordinary people, not from the government, not from wealthy donors, but from families who gave what they could, a dime at a time, because they believed that science could save their children. And in the end, it did. Into this landscape stepped two men whose approaches could hardly have been more different. Jonas Salk was a physician and researcher at the University of Pittsburgh who had worked on influenza vaccines during the war. He chose the killed virus approach.
Grow the polio virus in cell cultures, then inactivate it with formaldehyde. The formaldehyde would destroy the virus's ability to replicate while leaving its outer protein coat intact enough for the immune system to recognize. Show the body a dead virus, a corpse. Let it study the shape, build antibodies, form memory cells, and be ready when the living version appeared. The photograph in Salk's approach was a mugshot of a dead man. Safe, inert, but recognizable. Albert Sabin was a Polish-born virologist, older than Salk, more established, and by most accounts, forceful to the point of abrasive. He believed the correct approach was a live, attenuated virus. Take the polio virus, passage it through cell cultures repeatedly until it lost its ability to cause disease in humans, but retained its ability to replicate just enough to provoke a strong immune response. In Sabin's vision, the photograph wasn't a mugshot of a dead man, it was a live actor playing the criminal's role, convincing enough to train the guards, but unable to actually commit the crime. Sabin's approach had a theoretical advantage. Because his attenuated virus could still replicate in the gut, where poliovirus naturally establishes infection, it could produce stronger mucosal immunity. And it had a practical advantage. A live vaccine could be given orally, on a sugar cube. No needles, no trained medical personnel. You could set up a table in a village square and hand out sugar cubes. For developing countries where syringes and nurses were scarce, this mattered enormously. The rivalry between Salk and Sabin was intense and sometimes bitter. Sabin publicly called Salk's killed virus approach inadequate. Salk felt his work was being unfairly maligned. Beneath the scientific disagreements was something sharper. Sabin resented the public attention Salk received. There was a class dimension. Sabin came from the European academic tradition and viewed himself as a scientist's scientist. Salk was more of a public figure, willing to speak to journalists in ways that some peers considered unseemly. The personal dynamics between the two men were made worse by the way the public perceived them. Salk was handsome, articulate, and photogenic. He gave interviews. He appeared on the cover of Time magazine. He famously refused to patent his vaccine, saying in a television interview, when asked who owned the patent, "Well, the people, I would say. There is no patent. "Could you patent the sun?" It was a great line, and it made him enormously popular with the public. But it infuriated some of his scientific colleagues, who felt he was grandstanding. And it particularly infuriated Sabin, who viewed Salk as a technician rather than a true scientist, someone who had merely applied existing techniques rather than making fundamental discoveries. The truth, as usual, was more complicated.
Salk had made real intellectual contributions to the killed virus approach. His insistence that a properly inactivated virus could produce adequate immunity went against the prevailing orthodoxy, which held that only live viruses could generate lasting protection. He was right, and proving it required both scientific insight and personal courage. But the narrative that he was merely a technician who got lucky, promoted heavily by Sabin and others, stuck in many circles. Salk spent the rest of his career feeling under-recognized by his peers, even as the public adored him. But urgency demanded action. In 1954, Salk's vaccine was ready for what became one of the largest medical experiments in American history. The March of Dimes organized a field trial involving nearly 1.8 million children. These kids, mostly first through third graders, became known as the polio pioneers. Some received the vaccine, some received a placebo, some were observed as controls.
The logistics were staggering. Hundreds of thousands of doses manufactured, shipped cold, administered by thousands of physicians and nurses. The results were compiled by Thomas Francis Jr., Salk's former mentor, who insisted on rigorous independence. The announcement came on April 12, 1955, exactly 10 years after Roosevelt's death. Francis declared the vaccine safe, effective, and potent. Effectiveness between 60 and 90% against paralytic polio. Church bells rang, factory whistles blew, people wept in the streets. Jonas Salk became one of the most famous scientists in the world. The atmosphere in the auditorium was electric. Hundreds of scientists, public health officials, journalists, and March of Dimes volunteers packed the hall. Loudspeakers carried the announcement to crowds gathered outside. Across the country, people huddled around radios and televisions. Department stores piped the broadcast over their public address systems.
When Francis finished speaking and the word effective was confirmed, the reaction was instantaneous and overwhelming. People in the auditorium leapt to their feet. In stores and offices and homes across America, people cheered and cried. One mother was quoted saying, "I went to the nearest phone and called my husband, and we just cried together." School bells rang. Judges in courtrooms adjourned proceedings to make the announcement. It was a moment of collective relief so intense that people who lived through it would remember it decades later as one of the defining moments of their lives, alongside the end of World War II and the moon landing. Salk became an overnight hero. He was deluged with letters, more than he could ever answer. Children sent him drawings. Parents sent him prayers. One telegram read simply, "Thank you, thank you, thank you." The adulation was genuine and deeply felt, and it was fueled by years of accumulated terror that were now, at last, beginning to dissolve. And then, within weeks, something went horribly wrong. Qatar Laboratories, one of 5 manufacturers licensed to produce the vaccine, had batches where the formaldehyde inactivation process failed. The virus was grown in cell cultures that produced biological debris, and in some batches, clumps of this material had shielded pockets of live virus from the formaldehyde, tiny bunkers where the chemical couldn't reach. The virus in those vials wasn't fully dead. About 120,000 children received defective doses. Around 40,000 developed a mild form of polio,
56 developed paralytic polio. 5 children died. And because live poliovirus spreads through fecal-oral transmission, the damage rippled outward into families and communities. In total, the Qatar incident caused roughly 164 cases of paralysis and 10 deaths. The vaccination program was immediately halted. Congressional hearings were convened.
The investigation identified a manufacturing failure, not a flaw in Salk's design. The protocol worked, the principle was sound. But the margin for error was razor thin, and Qatar's quality control hadn't caught the problem. This moment reshaped how the United States government oversees biological products. New regulations were written. Manufacturing standards were tightened dramatically. Every batch of vaccine required more rigorous testing before release. The incident also raised questions about liability that would echo through decades, eventually leading to the National Childhood Vaccine Injury Act of 1986, which created a no-fault compensation system. The vaccination program resumed with stricter protocols, and it worked. Polio cases dropped from roughly 29,000 in 1955 to about 5,600 by 1957. Meanwhile, Sabin pushed forward with his oral vaccine.
He couldn't conduct large-scale trials in the United States because Salk's vaccine was already being widely used. So, at the height of the Cold War, the American virologist collaborated with Soviet researchers, particularly Mikhail Chumakov and Marina Voroshilova, to conduct massive trials involving tens of millions of people. The results were impressive. The oral vaccine was effective, easy to administer, and produced strong intestinal immunity that could interrupt community transmission. Sabin's oral polio vaccine was licensed in the United States in 1961 and gradually replaced Salk's injected vaccine in most of the world. But Salk's worry about reversion proved well-founded. In very rare cases, roughly 1 in every 2.4 million doses, the live virus mutated back toward virulence. It could cause the very disease it was designed to prevent. This was called vaccine-associated paralytic poliomyelitis. And for decades, as wild polio was driven to near extinction, a small but steady number of paralysis cases were caused by the vaccine itself. Once wild polio was eliminated from the United States, every new case of paralytic polio in the country was vaccine-derived. The tool built to fight the disease had become the only source of the disease. This is why the United States switched back to Salk's inactivated vaccine in 2000. The killed vaccine can't cause polio. It can't revert. In a country where wild polio no longer circulates, that trade-off makes clear sense. But in countries still fighting active outbreaks, the oral vaccine remained essential because of its ability to spread immunity through communities. Both vaccines turned out to be right, in different contexts, for different stages of the fight. The rivalry between Salk and Sabin was never resolved personally. Sabin remained dismissive of Salk until the end. Salk was never awarded the Nobel Prize, but their combined impact is almost beyond calculation. When the Global Polio Eradication Initiative launched in 1988, there were an estimated 350,000 cases of paralytic polio per year across 125 countries. By 2023, that number was down to single digits. We are closer to eradicating polio than any human disease except smallpox. And the tools that made it possible were invented by 2 men who could barely stand each other. And that's what makes the polio story so illuminating, because it shows every dimension of the vaccine challenge playing out simultaneously. The science of how to make a photograph, the engineering of how to manufacture it at scale,
the ethics of testing it on human beings, the politics of choosing between two competing approaches, the tragedy of what happens when manufacturing fails, the triumph of what happens when it works.
And the long, complicated question of when to switch from one approach to another as the landscape changes. There's a story that one of the most experienced vaccine scientists in America tells about this switching question. He joined the CDC's advisory committee specifically to push for the change from oral polio vaccine to the inactivated version, because by the 1990s, the only cases of polio in the United States were being caused by the vaccine itself. 7 to 10 children paralyzed by the oral vaccine every year, in a country where wild polio had been gone since 1979. And when he proposed the switch, the pushback was fierce, even from people he respected. One colleague pointed out that switching to the more expensive inactivated vaccine would cost about $4.5 million for every case of vaccine-associated paralysis prevented, as if you could put a price on a child's ability to walk.
He brought a vaccine safety activist onto the committee, a man whose son had been paralyzed by the oral polio vaccine and who had later died from the condition. This man wasn't an anti-vaccine activist. He didn't oppose vaccination. He wanted a safer vaccine. He wanted the inactivated version that Scandinavian countries had been using successfully for decades. And the committee members, who had been reluctant to make the switch, had to sit across the table from this father and explain to him why the change was too expensive. The switch eventually happened in 2000, and no child in America has been paralyzed by a polio vaccine since. But here's the complexity. That same oral vaccine, the one that was pulled from use in the United States because it caused a handful of paralysis cases each year, is still the primary tool for fighting polio in parts of Africa and Asia. Because in those settings, where wild polio still circulates and where the infrastructure for injectable vaccines is limited, the benefits of the oral vaccine, its ease of administration, its ability to spread immunity through communities, its low cost, still clearly outweigh its risks. The right answer depends on where you are, what you're facing, and what alternatives are available. There is no single right answer for all contexts, and this, perhaps, is the deepest lesson from the polio story. Medicine is never just about biology. It's about trade-offs, about weighing risks against benefits, about making the best decision you can with the information you have, knowing that the information is never complete. One of the most senior members of the FDA's Vaccine Advisory Committee, a man who co-invented a vaccine that has saved hundreds of thousands of children's lives worldwide, described the feeling of recommending a new vaccine for millions of people with a poker analogy.
You make your bet, he said, realizing you may have to tear up your ticket at the end of the race.
And doing nothing, he added, is also making a bet. The choice to not vaccinate is also a choice, with its own consequences. When the mRNA vaccines for COVID-19 came before his committee in December 2020, each member received about 400 pages of clinical data to review, from the company and from the FDA's own independent analysis. They had about a week to read every word. And they read every word because people's lives depended on whether they caught something in those pages that didn't look right. At that first meeting, they noticed a small signal. A statistically significant increase in Bell's palsy, a temporary facial paralysis, in the vaccinated group compared to the placebo group. The numbers were small, but the signal was there, and it worried them.
As it turned out, when the vaccine was given to millions of people and there was enough data on both sides, the signal disappeared. Bell's palsy wasn't a real side effect of the vaccine. But then came myocarditis, inflammation of the heart muscle, occurring primarily in boys and young men 16 to 29, primarily after the second dose, primarily within 4 days. At its peak in that demographic, the rate was roughly 1 in 6,600. That's not vanishingly rare, but it was almost always transient and self-limiting. The inflammation resolved on its own. Initially, affected patients were hospitalized, but as doctors gained experience, most cases were managed as outpatients. It was a real side effect, a genuine price paid by some people for the protection the vaccine provided. And the committee had to weigh that price against the protection, against the thousands of children under 18 who died of COVID,
against the 12-fold increase in hospitalization risk among the unvaccinated, and with the Johnson & Johnson vaccine, the adenovirus vectored vaccine, the price was higher. Severe blood clotting, including in the brain, rare, roughly 1 in 200,000, but real and sometimes fatal. That vaccine was ultimately pulled from the market. Because another option existed, because the mRNA vaccines were available and didn't carry that risk. The existence of an alternative changed the calculation, the same way the existence of the inactivated polio vaccine changed the calculation for the oral vaccine. Every decision is a bet, and the people making these bets know it. They carry the weight of it. Their hearts are in their throats, as one of them described it, every time a vaccine they helped recommend goes into millions of arms. They pore through databases asking themselves what they didn't think of. Are we causing an immune response against the cells that line your joints, against the cells in your pancreas that make insulin? Could we be creating diabetes? You don't think about everything because you can't think about everything. And eventually the vaccine goes out into the world and you find out what you missed. Through all of this, from Jenner to Pasteur to the great polio drama, the photograph was getting sharper, more precise, more refined. First, it was a living relative of the pathogen, then a weakened version of the pathogen itself, then a killed version, its shape preserved but its machinery destroyed. Each refinement made the photograph safer and more predictable. But there was one person who took this refinement further than anyone else, who developed more vaccines than any scientist in history, and whose name almost nobody knows. His story begins with a sick 5-year-old girl and a cotton swab at 2 o'clock in the morning. Maurice Hilleman might be the single most important vaccinologist in history, and yet most people have never heard of him. Over his career, he developed or substantially contributed to more than 40 vaccines, including those for measles, mumps, hepatitis A, hepatitis B, chickenpox, meningitis, and pneumonia. By some estimates, his work has saved more lives than the work of any other individual scientist in the 20th century. He grew up on a farm near Miles City, Montana, during the Great Depression. His twin sister died at birth. His mother died 2 days later. He was raised by an uncle and his uncle's wife. He worked at a J.C. Penney store to help pay for college. He earned a Ph.D. in microbiology from the University of Chicago, and from there he simply never stopped. Colleagues described him as blunt, demanding, sometimes intimidating. He had a relentless, practical approach to science. He didn't want an elegant hypothesis.
He wanted a vaccine that worked, that could be manufactured at scale, that would actually reach children. And the story of how he developed the mumps vaccine is one of those tales that feels almost too perfectly human to be real. In 1963, Hilleman's 5-year-old daughter, Gerald Lynn, woke him up in the middle of the night complaining of a swollen jaw. She had the mumps. Most parents would have given their child some soup, tucked her back in,
and sat with her until she fell asleep. Hilleman grabbed a cotton swab, went to his daughter's bedside, and swabbed the back of her throat. Then he drove to his laboratory at Merck, stored the sample, and began the long process of growing the virus, passaging it through cell cultures, weakening it step by step, generation after generation, until it could provoke an immune response without causing disease. The resulting vaccine strain is still called the Gerald Lin strain, named after his daughter.
It's still the mumps component used in the MMR vaccine, given to children around the world today. A vaccine born from a father's instinct at 2 a.m, from a sick girl's throat swab, and it's been protecting billions of people for over half a century. Gerald Lin Hilleman grew up fine. She became a successful businesswoman. And every child who gets an MMR vaccine carries, in a sense, her name. Hilleman's contribution to measles was equally important. The measles vaccine he helped develop became the standard by 1968. It's essentially the same vaccine still used today, which tells you how well he engineered it. But Hilleman didn't just develop individual vaccines. He was instrumental in creating combination vaccines that simplified childhood immunization. The MMR, measles, mumps, rubella, was his idea, licensed in 1971. Before MMR, children needed separate shots for each disease. More doctor visits, more needles, more chances for families to fall behind schedule and leave children unprotected. Combining 3 live attenuated viruses into a single injection was a significant engineering challenge. You had to ensure 3 different viruses could coexist in the same vial without interfering with each other, that they'd all remain stable, and that the immune response to each component would be strong enough. Hilleman and his team figured it out. And here's something that cuts through one of the most common concerns people have about childhood vaccines. 100 years ago, children received one vaccine, the smallpox vaccine, just one. But smallpox is the largest of the mammalian viruses. It has about 200 structural and non-structural proteins, 200 distinct molecular targets for the immune system to process. Today, children receive vaccines against 14 different diseases. The number 14 is bigger than 1, obviously.
But if you add up all the immunological components in all those modern vaccines combined, the total comes to roughly 150 proteins. Fewer than that single smallpox vaccine of a century ago. The photograph has gotten sharper, more focused, less cluttered. Thanks to advances in protein purification and recombinant DNA technology, modern vaccines present the immune system with a more precisely targeted challenge than their predecessors did. One of the lesser-known things Hilleman did happened in 1957 during the Asian flu pandemic. He was working at the Walter Reed Army Institute of Research, noticed reports of a new influenza strain emerging in Hong Kong, obtained samples, and realized this was a genuinely novel strain against which the population had no immunity. He sounded the alarm and pushed vaccine manufacturers to start production months before the pandemic wave hit America.
Because of his early warning, roughly 40 million doses of vaccine were available by the time the virus arrived that fall. The pandemic still killed about 70,000 Americans, but without Hilleman's intervention, the toll would have been far worse. He saw the wave coming and built the seawall before it hit shore. There's another story about Hilleman that deserves telling, because it shows the kind of relentless, unsentimental problem-solving that characterized his entire career. When his team was developing the hepatitis B vaccine in the late 1970s,
they faced an unusual challenge. The hepatitis B virus was extremely difficult to grow in cell culture. They couldn't mass-produce it the way they could measles or mumps. So where did the initial supply of viral material come from? From the blood of chronically infected individuals, many of them gay men in the New York City area, who were carriers of the virus.
Hilleman's team purified the viral surface protein from donated blood plasma, inactivated it thoroughly, and used it as the vaccine antigen. The timing was brutal. The vaccine was being developed just as the AIDS epidemic was beginning, and the blood supply was increasingly under suspicion. There were fears, unfounded but persistent, that the hepatitis B vaccine derived from blood might transmit HIV. It didn't. The purification and inactivation processes eliminated any risk. But the perception problem was real, and it contributed to a recombinant version of the vaccine being developed, one that used genetically engineered yeast cells to produce the hepatitis B surface protein, instead of extracting it from human blood. This recombinant vaccine, introduced in 1986, was one of the first vaccines to use recombinant DNA technology, a major milestone in the evolution of vaccine design, and it represented yet another step in the progression we've been tracking all night.
From living pathogen, cowpox, to weakened pathogen, Pasteur's attenuated bacteria, to killed pathogen, Salk's formaldehyde-treated poliovirus, to purified protein, the original hepatitis B vaccine, to recombinant protein, the yeast-produced hepatitis B vaccine, to genetic instructions, mRNA vaccines. Each step took the photograph further from the actual criminal. Each step reduced the risk. Each step increased the precision. And each step required a deeper understanding of the biology, a more sophisticated set of tools, and a willingness to rethink assumptions about what a vaccine could be. By the time mRNA vaccines arrived, the photograph had been abstracted so far from the original pathogen that it was no longer a photograph at all. It was a set of instructions for building a photograph. Your own cells received the instructions, built the image themselves, displayed it for the immune system, and then destroyed the instructions. The vaccine never contained any part of the virus, just the information needed to make one protein from its surface. Just the face in the photograph. Everything else, the body, the weapons, the ability to replicate and cause disease, was stripped away. All that remained was the identity, the face. And that level of precision, that ability to reduce a dangerous pathogen to a single protein and deliver the instructions for making it as a simple injection, is the endpoint of a 200-year journey that started with Jenna scratching cowpox into a boy's arm. From the crudest possible photograph, a living relative of the pathogen scratched into human skin, to the most elegant, a synthetic RNA molecule encoding a single protein, wrapped in a lipid bubble smaller than a wavelength of light. Hilleman died in 2005 at the age of 85. He never received a Nobel Prize, which, given what he accomplished, feels like one of the more notable oversights in the history of that award. But in vaccinology, his legacy is unquestioned. And yet, for all the progress Hilleman represented, every one of his vaccines still required the actual pathogen at some point. You had to grow the virus, handle it, weaken it or kill it, or extract proteins from it. The raw material was always the pathogen itself, and that requirement carried inherent limitations. Growing viruses takes time. It requires specialized facilities,
It involves handling dangerous organisms. And for a brand new pathogen, one that has just emerged and is spreading rapidly, the time it takes to isolate, characterize, grow, and develop a vaccine can stretch to years. So the ultimate dream was to find a way to skip the pathogen entirely, to not need the virus at all, to take only the information, the genetic blueprint, the instructions for building the photograph, and let the body do the rest.
The technology that would make this possible had been quietly developing for years, championed by a handful of researchers who believed in it, when almost nobody else did. In 1997, at the University of Pennsylvania, a biochemist named Katalin Karikó was having a difficult career. She had been working on messenger RNA for years, and her work was going nowhere in terms of professional recognition. Her grant applications were being rejected. Her department had denied her a promotion. She'd been demoted, actually, losing her position on the tenure track. The problem wasn't her intelligence or her work ethic. The scientific establishment didn't believe in what she was trying to do. Her idea was to use synthetic messenger RNA as a therapeutic tool, to inject mRNA molecules into the body and have cells use them as instructions to build specific proteins. If it worked, you could theoretically produce any protein you wanted inside the body just by delivering the right mRNA.
The applications would extend far beyond vaccines. But there was a problem. When you inject synthetic mRNA into the body, the immune system recognizes it as foreign and attacks it. Those pattern recognition receptors we talked about at the very beginning of tonight's story, those toll-like receptors, can detect structural differences between your own mRNA and foreign mRNA. The synthetic material triggers an inflammatory response. The body treats it as a threat and destroys it before it can do any useful work. And here's where something beautifully ordinary happened. Karikó was standing at a copying machine in the hallway of her department one day, and she struck up a conversation with another researcher named Drew Weissman. He was an immunologist who had studied under Anthony Fauci at the National Institutes of Health. He was interested in developing better vaccines, particularly for HIV. They started talking. They kept talking. And eventually they began collaborating on the problem that had been blocking Karikó's progress for years. How do you get synthetic mRNA past the immune system's defenses? Their solution, published in 2005, was elegant. They discovered that by modifying one of the 4 nucleoside building blocks of mRNA, replacing uridine with a slightly altered version called pseudo-uridine, they could make the synthetic mRNA essentially invisible to the innate immune system. The toll-like receptors that would normally detect foreign RNA and trigger inflammation simply didn't recognize the modified version. The mRNA survived. The cells took it up. They read the instructions. They built the protein. And the inflammatory response was dramatically reduced. It was a fundamental breakthrough, and at the time, almost nobody noticed. The paper attracted little attention. The scientific community remained skeptical of mRNA as a therapeutic platform. Karikó and Weissman kept working, refining their technology, publishing papers that accumulated quietly. Years passed. Meanwhile, a few small companies began to see the potential. Moderna, founded in 2010, was built specifically around modified mRNA technology. BioNTech, a German company founded by Ugur Sahin and Özlem Türeci, was pursuing similar ideas. These companies were investing in mRNA before most of the world knew it existed. And then came January 2020. On January 10th, Chinese scientists published the full genetic sequence of a new coronavirus, SARS-CoV-2. They posted it online, an electronic file, a string of letters, AUGC, roughly 30,000 of them, representing the complete blueprint of the virus. Within days, scientists at Moderna in Cambridge, Massachusetts, had designed an mRNA vaccine candidate. They didn't need the virus itself. They didn't need to grow it, handle it, kill it, or weaken it. They just needed the sequence.
The information. The genetic code of a single protein, the spike protein on the surface of the coronavirus, the protein the virus uses to latch onto human cells. They took that sequence, synthesized the corresponding mRNA with the modified nucleosides that Carrico and Weissman had pioneered, wrapped it in a tiny lipid nanoparticle, a fat bubble to protect it during injection and help it enter cells, and the vaccine candidate was essentially designed. The early history of mRNA research is itself a story worth knowing, because it shows how close the whole enterprise came to being abandoned before it ever got started. In 1989, a researcher named Robert Malone, working at the Salk Institute no less, demonstrated that you could take messenger RNA, inject it into mouse muscle cells, and the cells would take up the mRNA and produce the encoded protein. It was a proof of concept, a laboratory demonstration that the basic idea could work in living tissue.
But it was a long, long way from a usable medical product. The problems were formidable. Synthetic mRNA was fragile. It degraded rapidly. The immune system attacked it. The protein production was fleeting and inefficient. Getting mRNA into cells in a living animal, rather than in a dish, was enormously difficult. And the inflammatory response it provoked was so severe that many researchers concluded the whole approach was a dead end. Funding dried up. Interest waned. Malone himself moved on to other work and would later, in an ironic twist that mirrors Andrew Wakefield's trajectory, become a vocal critic of the very technology he had helped pioneer, speaking out against mRNA vaccines during the COVID pandemic. But Carrico didn't give up. Through the 90s and early 2000s, when mRNA was considered a backwater of biomedical research, she kept working. She kept losing grants. She kept getting passed over for promotions.
She kept trying different modifications to the mRNA molecule, searching for the one that would make it invisible to the immune system's defenses. And when she and Weissman finally found it, the pseudo-uridine modification, in 2005, they had solved the central problem that had defeated everyone else. They had figured out how to whisper to the cell without waking the guards. It took another 15 years for the technology to reach its full potential, and it required a global pandemic to create the urgency and the funding for the fastest vaccine development program in history. But the scientific foundation had been laid years earlier, quietly, painstakingly, by researchers who believed in the work when almost nobody else did. That gap between discovery and application, between the laboratory breakthrough and the real-world product, is one of the recurring themes of this entire story. Jenner waited years for acceptance.
Pasteur spent decades building from crystals to vaccines. Hilleman worked for a quarter century on rotavirus, and Carrico worked for over 20 years on mRNA before the world finally needed what she had built. By February 24th, Moderna shipped the first batch to the National Institutes of Health for testing. By March 16th, the first human volunteer received an injection. The design-to-first injection timeline was 66 days. No vaccine in history had ever moved that fast. And the speed wasn't from cutting corners. The mRNA technology had been in development for over 2 decades. The clinical trials that followed were large, rigorous, and conventional. 40,000 participants for Pfizer's trial, 30,000 for Moderna's. What was eliminated was the dead time, the years of growing virus, the years of manufacturing development. Think about the contrast with the Balmis expedition. In 1803, to transport a vaccine across the Atlantic, a physician loaded 22 orphan boys onto a ship and infected them sequentially, arm to arm, child to child, to keep the living virus alive during the voyage. 217 years later,
To get a vaccine to the other side of the world, someone sent an email attachment. A text file. 4 letters. 1 gene. The photograph had become a text message. Carrico and Weissman were awarded the Nobel Prize in Physiology or Medicine in 2023. The copying machine where they met is still in the hallway at the University of Pennsylvania. Let me explain what actually happens inside your body when an mRNA vaccine does its work, because the process is a beautiful illustration of everything we've talked about tonight. You receive an injection. The lipid nanoparticle fuses with the membrane of some of your cells, typically muscle cells near the injection site.
The mRNA is released into the cell's cytoplasm, the fluid-filled space where the cell's protein-making machinery operates. The cell's ribosomes, the molecular machines that read mRNA and build proteins, encounter the synthetic mRNA and do what they always do. They read the instructions and build the protein. In this case, the spike protein of the coronavirus. That spike protein by itself can't cause COVID. It's just one protein, disconnected from the rest of the virus, with no genetic material, no ability to replicate. But it's foreign. It doesn't belong in your cells, and your cells have a built-in mechanism for dealing with foreign proteins. They chop them into fragments and display those fragments on their surface, held up on special molecules called MHC proteins. Like a flag, like a sign that says, "Look what I found inside." Remember earlier when I described macrophages eating pathogens and displaying fragments on their surface? The same principle is at work here.
The cell is presenting a piece of the enemy's face to the immune system, showing the photograph, and your immune system responds. The hunters, those B cells and T cells with their randomly generated receptors, begin to encounter the spike protein fragments. Somewhere in that vast army, there are a few whose receptors match. They recognize the spike protein. They activate, they divide. Clonal expansion begins. Affinity maturation follows. Memory cells are formed. The synthetic mRNA itself doesn't last long. It's fragile by design. Within hours to days, the cell's natural enzymes break it down. There is no permanent alteration to the cell. The mRNA never enters the nucleus. It never touches your DNA. It does its job, and then it's degraded and recycled. But the immune memory persists. Your body has seen the photograph. It has trained its hunters. And if the real coronavirus shows up, the recognition is fast. Trained B cells produce antibodies that block the virus from entering cells. Trained T cells destroy cells that have already been infected. The response is overwhelming. That's how a vaccine works. The specific technology has evolved enormously, from cowpox scratched into an arm to synthetic mRNA wrapped in lipid nanoparticles. But the fundamental principle that Jenner glimpsed in 1796 has remained the same. Show the immune system a photograph. Let it learn. Let it remember. And there's one more aspect of the mRNA vaccine story that deserves attention, because it addresses what was perhaps the most common criticism leveled against these vaccines: that they were developed too fast, that the speed itself was evidence of recklessness or corner cutting. The speed was real, and it was genuinely unprecedented. But the explanation isn't that safety steps were skipped. The explanation is that every other vaccine in history had been slowed down by 2 bottlenecks that mRNA technology eliminated. The first bottleneck was biological.
Growing a virus takes time. Isolating it, characterizing it, figuring out how to culture it in large enough quantities to manufacture a vaccine. For some viruses, this process alone can take years. With mRNA, you don't need the virus, you just need its genetic sequence.
A string of letters posted on the internet. The biological bottleneck vanishes. The second bottleneck was financial. Normally, vaccine development proceeds in stages, and each stage depends on the previous one succeeding before the next one begins. You do phase 1 trials, small safety studies, wait for the results, and only then begin phase 2 trials, larger studies testing dosage and immune response. You complete phase 2, analyze the data, and only then move to phase 3, large-scale efficacy trials with tens of thousands of participants.
And you only build manufacturing facilities after phase 3 succeeds, because building a factory to produce a vaccine that might not work is an enormous financial risk that no company would normally take. What Operation Warp Speed did, the $11 billion program launched by the United States government, was take all of that financial risk away from the companies. The government paid for phase 1, phase 2, phase 3 trials, all overlapping. It paid for manufacturing facilities to be built before the trials were finished. It pre-purchased hundreds of millions of doses. If the vaccines hadn't worked, all of that money would have been wasted. The companies bore no financial risk, and because the stages overlapped rather than proceeding sequentially, years of dead time were eliminated. The science itself wasn't rushed. The trials were the same size as standard vaccine trials. The safety monitoring was, if anything, more intense than usual, because the whole world was watching. One of the scientists on the FDA advisory committee called Operation Warp Speed the greatest scientific and medical accomplishment of his lifetime. A virus with unusual characteristics met a technology, messenger RNA, that we had no previous experience with in the vaccine context, and the result was a safe and effective vaccine. The technology itself had been in development for over 20 years. The specific vaccines were tested in trials of 30 to 40,000 people. The post-authorization surveillance systems tracked hundreds of millions of doses. The speed came from eliminating financial risk and running processes in parallel, not from cutting corners on safety. And yet, the speed itself became a source of suspicion. People heard, "fastest vaccine ever developed," and translated that into insufficiently tested. Which is understandable, honestly. When your entire experience of product development tells you that faster means less careful, it's natural to apply that mental model to vaccines. But in this case, the analogy is misleading. The speed came from money and coordination, not from reduced rigor. And speaking of memory, there's one more piece of this story that I think is worth exploring before you drift off, because it answers a question you might have been carrying with you. A question with enormous practical implications. Here's something that puzzled people for a long time. If you had measles as a child, you are protected for the rest of your life. 60 years later, 70 years later, your immune system still remembers. There was a remarkable study on a remote island that had experienced a measles epidemic. Then, for about 40 years, the virus completely disappeared from the community. No measles at all for 4 decades. When the virus was reintroduced by visitors from abroad, the younger people, who had never been exposed, got sick. But the older people, those who had survived the original epidemic 40 years earlier, were completely protected. Their memory cells were still there, still functional, still ready. Compare that with COVID. People who recovered from COVID, or who were vaccinated, could get infected again within months. Their antibodies waned. Their protection against mild illness faded. They were still protected against severe disease, against hospitalization and death, but they were clearly vulnerable to reinfection in a way that measles survivors simply weren't. Why the difference? And the answer turns out to be one of the most important and underappreciated concepts in vaccine science. The key is the incubation period, the time between when a pathogen enters your body and when it causes symptoms. Measles has a long incubation period.
roughly 7 to 14 days from the moment the virus enters your respiratory tract to the moment you develop symptoms. That's a long time in immunological terms. And here's why it matters. Those memory cells, the B cells and T cells that survived after your first encounter with measles, they need time to reactivate. When they encounter the measles virus again, they don't produce antibodies instantly. They need a few days to wake up, start dividing, ramp up antibody production, and mount a full response. Measles gives them those days. The virus is slow to cause trouble. Those 7 to 14 days of incubation provide a window, a grace period, during which memory cells can reactivate, produce a flood of high-affinity antibodies, generate killer T cells, and mount a defense faster, stronger, and more overwhelming than the original response. The virus never gains enough of a foothold to cause illness. You don't even know you were re-exposed. Your immune system handled it behind the scenes during that long incubation window. Now consider COVID. SARS-CoV-2 has a short incubation period, sometimes as short as 2 to 3 days. The virus moves fast. It establishes itself in the upper respiratory tract and begins causing symptoms quickly before memory cells have had time to fully reactivate. The result is that even people with strong immunological memory can develop mild to moderate symptoms upon re-exposure. Their memory cells eventually kick in, and that's why the disease is much milder the second or third time. It's why vaccinated people are far less likely to end up in the hospital. The memory is there. But the protection against any infection at all fades as circulating antibodies decline over months, and memory cells can't respond fast enough to prevent mild illness, given how quickly the virus acts. This distinction has profound implications. For long incubation period diseases, vaccines can provide lifelong protection and potentially prevent transmission entirely, which means you can eliminate the disease. Smallpox, eradicated. Measles, eliminated from many countries. Polio, on the verge of global eradication. For these diseases, vaccination can break the chain of transmission completely. For short incubation period diseases like COVID or influenza, vaccines can still prevent severe disease and save enormous numbers of lives. But they're unlikely to prevent mild infection or transmission entirely. The virus keeps circulating. The goal of vaccination shifts from eradication to harm reduction. And there's another layer worth knowing about. In the late 1940s, a virologist named Thomas Francis described a phenomenon called original antigenic sin, now more commonly called immune imprinting. The idea is that the first influenza virus you encounter as a baby or young child leaves a lasting imprint.
Your immune system builds its strongest memory against that specific strain. And for the rest of your life, every time you encounter a new influenza strain, your immune system responds as if it's still fighting the first one. It preferentially activates those original memory cells, even when the new strain is different enough that those old antibodies aren't the best fit. Francis reportedly said he could tell approximately when a person was born based on how they responded to different influenza strains. Your immunological autobiography is written in the antibodies you carry. The implications of imprinting extend beyond influenza. Researchers are now studying whether similar phenomena occur with other pathogens, including coronaviruses. If your first exposure to a coronavirus was the original Wuhan strain of SARS-CoV-2, your immune system may have built its primary memory around that strain's spike protein.
When new variants emerged, Omicron and its descendants, your immune system's response may have been shaped, for better and for worse, by that original encounter. The boosted immune response might have targeted features shared between the original strain and the variant, which would help. But it might also have been slower to recognize and respond to the features that were genuinely new, which would not. This is an active area of research, and the answers are still emerging. But the concept itself, that your immune history shapes your immune future, that the sequence of exposures matters and not just the fact of exposure, is one of the most important ideas in modern immunology. And it has practical implications for vaccine design. Should booster vaccines be formulated to match the latest circulating variant? Or should they contain a mixture of antigens to broaden the immune response? How do you update a vaccine for a virus that keeps changing?
These are questions that vaccine designers are wrestling with right now, and they don't have easy answers. What we do know is that the immune system is not a simple on-off switch. It's not a matter of being immune or not immune, protected or unprotected. Immunity is a spectrum shaped by genetics, by age, by the specific pathogens you've encountered, by the order in which you encountered them, and by the vaccines you've received. Two people can get the same vaccine and develop different levels of protection because their immune histories are different, their genetics are different, and the complex interplay between innate and adaptive immunity unfolds differently in each individual. And yet, across all of this complexity, the fundamental principle holds. Showing the immune system a photograph of the threat prepares it to respond. The details of how well it responds, how long it remembers, and how effectively it protects depend on 100 variables. But the core strategy, the strategy that humans stumbled onto thousands of years ago and have been refining ever since, remains as sound today as it was when Chinese practitioners first blew scab powder into a child's nostril and watched, hoping, waiting, to see if the child would survive. The question of how long vaccine-induced memory lasts connects to something many people wonder about. Why do some adults who were vaccinated against measles as children show low antibody levels in blood tests? Does that mean the vaccine has worn off? Usually no. Circulating antibodies naturally decline over time. What matters is whether the memory cells are still there, still capable of reactivating if needed. And for most people, they are. Memory B cells and memory T cells can persist for decades, even when antibody levels have dropped to undetectable.
Testing for memory cells requires specialized techniques not used in routine medicine. But research consistently shows that immunological memory from childhood vaccination persists far longer than antibody levels alone would suggest. And there's one more concept worth understanding, because it explains not just individual protection, but how vaccination protects entire communities. Community immunity, sometimes called herd immunity, is the idea that if enough people in a population are immune, the pathogen can't find enough susceptible hosts to sustain transmission, and it fizzles out. Even people who aren't immune, newborns, people with compromised immune systems, the elderly, get indirect protection because the virus simply can't reach them. The threshold depends on how contagious the disease is. For measles, which is breathtakingly contagious, you need about 93 to 95% of the population to be immune. That's an incredibly thin margin.
If vaccination rates drop even a few percentage points below that, measles finds its way back. And we've seen this happen repeatedly in communities where vaccine uptake dropped. And measles deserves special attention here because it's the disease that most clearly illustrates both the power of vaccines and the fragility of the protection they provide at the population level. The measles virus is breathtakingly contagious, arguably the most transmissible pathogen humans have ever encountered. If you're not immune and you walk into a room where someone with measles coughed 2 hours ago, when the room is empty and the person has long since left, you can still catch it.
the virus hangs in the air, suspended in tiny droplets, lingering in enclosed spaces, waiting for an unprotected respiratory tract. The basic reproduction number for measles, the average number of people one infected person will infect in a fully susceptible population, is somewhere between 12 and 18. Compare that to influenza, where one person typically infects 2 or 3 others, or to the original strain of SARS-CoV-2, where the number was roughly 2 to 3. Measles is in another league entirely, and because measles is so contagious, the threshold for community immunity is extraordinarily high. You need roughly 93 to 95% of the population to be immune before the virus can no longer sustain transmission. That means if even 5 to 7% of people are susceptible, measles can find them. It's like a fire that can reignite from the smallest ember. We've seen what happens when vaccination rates drop below that threshold. In 2019, in Samoa, a tragic sequence of events demonstrated the consequences with terrible clarity.
The country's measles vaccination program had been disrupted after 2 infants died following MMR vaccination. Not from the vaccine itself, but from a catastrophic error in which nurses inadvertently mixed the vaccine with a muscle relaxant instead of sterile water. 2 babies died from the medication error, not from the vaccine. But the tragedy, understandably, shattered public confidence. Vaccination rates plummeted. Within months, measles swept through the country. Over 5,700 cases, 83 deaths. Most of the dead were children under 5. For a disease we have a safe, effective, inexpensive vaccine against, available for over half a century, those deaths are difficult to process. Each one represents a failure, not of the vaccine, which works extraordinarily well, but of the systems and trust and communication that are supposed to get the vaccine to the people who need it.
The man who developed the measles vaccine that's still in use today, Maurice Hilleman, might have had something characteristically blunt to say about it. He didn't suffer fools. He didn't tolerate complacency. And he understood, perhaps better than anyone, that a vaccine sitting in a warehouse is no different from no vaccine at all. The science is necessary, but it's not sufficient. You also need logistics, cold chains, trained healthcare workers, public trust, and the political will to maintain immunization programs year after year, decade after decade, even when the diseases they prevent have become so rare that people forget why they were vaccinated in the first place. That forgetting is one of the cruelest ironies of successful vaccination. The very success of vaccines, the disappearance of the diseases they prevent, undermines public motivation to keep getting them.
When parents have never seen a child with measles, never seen the rash, the fever, the encephalitis, the deafness, the death, the urgency of vaccination feels abstract. It feels optional. And that sense of optionality is what allows vaccination rates to slip below the threshold. And when they slip, the diseases come back. Every time. Without exception. For COVID, with its short incubation period and the ability of vaccinated people to still transmit, classic community immunity was never achievable. The goal was always about reducing severe disease. Both goals, eradication and harm reduction, are enormously valuable, but they serve different purposes, and understanding the difference helps make sense of why your childhood measles vaccine has lasted decades, while you might benefit from a COVID booster annually.
And perhaps the most important lesson from the COVID vaccine experience, the one that public health officials themselves acknowledged they communicated poorly, is the difference between what different vaccines can and can't do. When the COVID vaccines were first authorized, there was a narrative, partly driven by hope, partly by the urgent need to get people vaccinated, that these vaccines would stop the pandemic, that they would prevent infection, that vaccinated people wouldn't get sick. And for a few months, when antibody levels were high and the virus hadn't evolved much, That was roughly true. The protection against any infection was strong, but it was always going to be temporary. Because COVID is a short incubation period, mucosal infection. The virus attacks the respiratory tract, the surfaces of the nose and throat, and it moves fast. Circulating antibodies, the ones that provide protection against mild disease, decline naturally over months. That's normal.
That's what antibodies do. And once they've declined below a certain threshold, the virus can establish a foothold in the respiratory tract and cause mild symptoms before memory cells have time to fully activate and clear the infection. The memory cells, though, and this is the crucial point, do protect against severe disease. They protect against hospitalization. They protect against death. Because severe disease takes longer to develop than mild disease. The virus has to spread from the upper respiratory tract into the lungs, and from there, potentially, into other organs. That deeper invasion takes time. And that time is enough for memory cells to activate, for antibodies to be produced, for killer T cells to destroy infected cells, and for the immune system to bring the infection under control before it becomes life-threatening. So the vaccines worked. They worked magnificently for what they were biologically capable of doing. They prevented vast numbers of hospitalizations and deaths. But they couldn't prevent every sniffle and cough, because no vaccine against a short incubation period respiratory virus can do that. Not the flu vaccine, not any future COVID vaccine. The biology doesn't allow it. And the failure to communicate this distinction clearly, the failure to set realistic expectations about what the vaccines could and couldn't do, eroded trust. People got vaccinated, were told they were protected, and then got COVID. They felt betrayed. They felt lied to. And some of them concluded that the vaccines didn't work at all, which is wrong, tragically wrong, but emotionally understandable. The vaccines kept millions of people alive who would otherwise have died. But the messaging oversold what they could achieve, and the gap between expectation and reality became a breeding ground for distrust. If the story of vaccines teaches us anything, it's that honesty about limitations is just as important as enthusiasm about achievements. Jenner's vaccine worked, but it needed quality control. Pasteur's vaccines worked, but his backstory was messier than his public story. Salk's vaccine worked, but the cutter incident showed how thin the manufacturing margins were. Sabin's vaccine worked, but the reversion problem was real. The mRNA vaccines work spectacularly well at what they're designed to do. But they can't do everything. No vaccine can. And saying so honestly, clearly, and from the beginning is part of what makes the whole enterprise trustworthy. Because trust is the foundation. Without trust, the most brilliant vaccine in the world sits unused in a warehouse. And trust isn't built by over-promising. It's built by explaining honestly what you know, what you don't know, and what you're doing to find out. It's built by acknowledging the risks and showing the systems you've put in place to monitor them. It's built by telling the whole story, the messy parts included, and letting people see the humanity behind the science. Which, in a way, is what we've been doing tonight. Your immune system does its best. It always does. And vaccines simply give it a head start, showing it the photograph before the intruder arrives. How much head start it needs, how detailed the photograph must be, and how long the memory lasts, all of that depends on the dynamics of the pathogen and the immune response. And the story of how humans figured this out, from practitioners blowing scab powder into nostrils to researchers sending genetic sequences by email, is one of the longest, most winding, most consequential journeys in the history of medicine.
So here we are. And maybe you're still fully awake, following every word.
Or maybe you've been drifting in and out, catching phrases here and there, letting the details blur while the larger shape of the story settles into place. Either way is fine. The story doesn't require your full attention to do its work. Because right now, as you lie here, everything we've talked about is happening inside you, quietly, without any effort on your part. Your innate immune system is patrolling. Macrophages are stationed at every border. Neutrophils are circulating, ready to respond in minutes. Your toll-like receptors are feeling for the ancient signatures of infection, the patterns that evolution taught your body to recognize hundreds of millions of years ago. And your adaptive immune system, that extraordinary army of hunters, is out there too. Billions of lymphocytes, each one unique, each one waiting for its specific target. Most of them will never find it. Most will live their entire lives without encountering the one pathogen they were built to fight. But they're there, patient, ready, waiting for a creature that may never come. And scattered among them, in your bone marrow, in your lymph nodes, in the quiet tissues of your body, are the memory cells, the veterans. They carry the imprint of every vaccine you've ever received, every infection you've ever fought. Measles, mumps, rubella, tetanus, maybe COVID, maybe influenza from last winter. They remember. They've refined their weapons through affinity maturation. They've sharpened their receptors. They've trained themselves to recognize their targets with a precision that no technology has ever matched. And if any of those targets ever appear again, the memory cells will activate in hours rather than days, mounting a response so fast and so complete that you'll never know the battle happened.
The whole arc of what we've talked about tonight, from variolation in ancient China to Lady Montagu's fierce advocacy in London, from Jenner scratching cowpox into a boy's arm in a country garden to Pasteur's forgotten bacterial cultures, from Salk's killed virus to Sabin's sugar cubes, from Hilleman's 2 a.m. throat swab to Carico's quiet years at the copying machine, all of it comes down to one idea. Show the immune system a photograph. Let it study the face. Let it build its army. Let it remember. 200 years ago, delivering that photograph across an ocean required a ship with 22 children, passing a living virus arm to arm through the waves. Today, it takes a text file, 4 letters, sent at the speed of light, and somewhere on the other end, a laboratory begins making a vaccine within hours, without ever touching the pathogen, without ever being in the same room with the disease. The photograph has become a message. The message has become a memory. And the memory lives inside you right now, doing its work, standing guard, keeping you safe while you rest. You don't need to hold on to any of the details. You don't need to remember the names or the dates or the molecular mechanisms. Your body remembers for you. It's been remembering since before you were born, and it will keep remembering long after you've forgotten tonight's story. So let it do its work. Let the hunters patrol. Let the memory cells keep their vigil. Let your innate defenses scan the borders one more time and find nothing, and report back that everything is quiet, everything is safe, everything is exactly as it should be. The photograph is filed, the guards are posted, and you can rest. Just breathe and sleep.